Do we have any audio now? Okay, great. We have some amount of audio so we can get going. All right, just let me shut down the other live that's going on so that there'll be no confusion. Good morning and welcome to CSEC Biology TCP. We are on our regular program, the live show called Mr. Wilson Live. I am Mr. Wilson from the TCP Academy, your preferred place for online biology and HSP classes. We are going to be doing the final touch for the exam. We're going to really crave the support of those students who are better prepared, and also those teachers who would have the time to either join as a ghost teacher or otherwise. We really need all the support that we need getting into the exam. It's now 9 minutes after 8, and we intend to take you as close as possible to the exam. I did log on earlier and I did give some pointers as to some areas that you could cover. I do hope that we were able to do that. If we were, then we are almost good to go. I did tell you that my stock would have been expended, so I'm just going to try to write from the back of the past paper for the things that we are supposed to cover. Of course, we are only covering those things that are on the broad topic for the exams. Now, I know that everybody is anxious, including Mr. Wilson, who is not sitting the exam. And my son is not sitting the exam either. But I am as anxious as you are and probably excited at the same time. It is indeed my pleasure to have served for a year. The SWOT analysis, the strength. I have had the opportunity to meet students and teachers from right across the Caribbean. In a matter of fact, more than the Caribbean. I have had the opportunity to access resources that were not normally available to me. And these resources came to me from teachers, came to me from students. Have I grown as a person? Yes, I think I have grown. I think I have seen the content a little different. I think I have a greater appreciation for the subjects and the different tiers of learner. Yes, you're teaching at one place, so you pretty much have sort of one modality. But teaching on the platform, I get the opportunity to meet adults, kids, those who are really bright, those who are in between, and those who are struggling somewhat. So those are some of the strength, some weakness. There were times when the system fell apart. There were times, not many times, when we were intercepted by persons promoting stuff or pornography. In very few cases, we might have had a student who went overboard. Weakness, I am not able to give you that quality presentation that I'd love to have given. I would have loved to have had a chalkboard and cameras and proper lighting and proper software that I can share my screen, software that is not breaking down and you have to take another extra 10, 20 minutes before you can get order. Those were some real weakness. Having adequate resource that I can reach all my learner, teaching aids and so on, that was not available because of a limited budget that we don't have a budget. I really teach them on a budget, right? So the strength, weakness. What are the opportunities? The opportunities exist if we could get a sponsor for any of the program that we offer because we love to continue teaching online. The reality is some students, some persons just really cannot afford it. And you have persons who are really, they have the potential, but they need that help. So if the private sector could pretty much assist us, and from time to time we could have like guest teachers and so on coming on to make the product a little better. Those are some opportunity. The opportunity, I might fly to all these Caribbean one day and meet my students, and I don't know what, promote my book. I have written, I think I like COVID more than anything else. I have gotten the opportunity to grow as an individual. I read a lot, I watched a lot, I wrote a lot, I reach out to a lot of kids reach out to me. So those are really good for me. Exam starts at 1 p.m. in Jamaica. Yes. Great. So we are at strength, weakness, opportunities. Now the threat. The threat is somewhat good and somewhat bad. Now, when I was with Amoy and the team, when we probably had about 20 odd students, I could just call on Amoy or call on Tanisha or call on Macmillan or call on DeSouza's, call on Barnett. I don't want to start calling names, and I know exactly who was there and I know who didn't come to class today and who should have been at class and who is late for class and why are you late for class. It was like I knew these kids, right? And as the channel grow, I struggle to find them online. I have to be told from the back end who are the persons who were there because I have to really continue. I am really concerned about these kids that I've been teaching for over a year. So if they are not here, it troubles me. So that is a threat, not being able to reach everybody as an individual because the room is so big now. The scroll is so fast now. But my assistant in the background, she pretty much keeps a check of what is happening. She knows just about everybody that is on the channel. So while I am teaching and I can't see here, usually I get a report as to some things that I would have missed in the chat and so on, right? So that is a threat. Another threat is that we will not be able to continue 3 shows come September. We will not be able to do that unless we have a budget or we are financed to do that sort of thing. We can almost pledge that we're going to be having at least 1 show. As it were, for exam and marathon, once I'm available, I am going to be here doing that with whatever student, wherever they are from. We are going to continue to put out videos. Yes, we are going to continue to put out several videos, and we will try as much as possible to have a minimum of 1 show per week. Usually the 1 show is 2 hours, or we might make it 2 1 hour, that sort of a thing. But I don't know that we can continue to really do 3 shows per week. It's really taxing. We're going back to school, and I have school to do and all of that, plus the show. So that is a threat to the show. It doesn't mean that we are going to be giving anything less. For those in Jamaica, the plus for it, well, if you are in my domain, how we can make up with that, I have an online platform. So persons who can afford it, you can join me over there if you really need the help. It's going to be really affordable, so no need to worry. Sir, I have a class that I'm teaching and I am sitting the exam. We have it at 11 a.m. today. Fuller, you have a class. Well, I'm sorry, but If you can't be here, you just can't be here. There are some things that we have to do. We are creating a brand, and there are some things that we do at a particular time. So I'm really, really sorry. You can catch a replay or catch a video, that sort of a thing. I probably don't understand what you're saying. I think you're saying that get on with it because I have a class. I don't know if that is what you're saying, but if that's what you're saying, I am saying that I'm sorry, but you can catch a replay if you can't be here. I do understand. All right, so we want to hop right into the show. And to get into the show, I want to remind those persons who are not familiar with what is expected of them today that on the paper, we're expecting to see living organisms and the environment. That takes in the entire section A of the syllabus. We also note that the skeletal system should be on the paper, and we have some past paper that we recommended earlier that should be able to assist persons. Excretion and homeostasis is definitely going to be on the paper, and the chronic diseases that are associated with that. Reproduction, the reproductive system is going to be there. Heredity and variation, and disease and its impact on human. So we're going to be looking at things like a little bit on malaria. We're going to be looking at sickle cell anemia. Those are some stuff that we want to look at. We're going to be, I'd ask you to look at impact of health practice on the environment, and we're supposed to link it to infectious disease. Based on the syllabus I sent you earlier, that the syllabus suggests that you are supposed to go to D7. So the diseases that are in D7 of the syllabus are pretty much the diseases that we're expecting to be on the exam based on the broad topic and based on the link to the syllabus. With that said, I am going to start the lesson this morning with mitosis. And the syllabus did say that there is no need for you to know the stages, but I strongly disagree. So I'll be asking, and persons are going to be answering questions in the chat. And where possible, I'll just do a little sketch on my paper that persons can see. So I'll just lay the ground thing for mitosis. Mitosis is pretty much a type of cell division that takes place in autosomal cell. Now, what are autosomal cells? Autosomal cells are all the cells in the body except the sex cells. Now, your sex cells are going to be the sperms and the ovum. Now, why do we want to have mitosis? Why is mitosis important? It's probably something that persons are asking, being that last night persons didn't know what mitosis is. Now, mitosis ensures that the diploid number of chromosomes are pretty much passed on. What does that mean? The full number of chromosome is passed on. But why is that important? For example, if you were supposed to get a cut, you have to make new cells. So you want to make sure that those cells have the same number of chromosomes. While you grow, there are cells that are dying, there are cells that are being replaced. And you don't want to know that you have a baby today and the baby was nice and dandy. And by the baby reached 2 year old, the baby is a turtle. And then when it reached 3 year old, it becomes a crocodile. And then 5 year old, it becomes an iguana. You really don't want to do that. So you want to ensure that the chromosome numbers are maintained. And as such, that's one of the importance of mitosis. Those are not the only importance of mitosis. It makes sure that the genes, so to speak, are identical identical. So each time you see your child, you know that that is your child, because even though the child is growing, the cells that are replaced, the chromosome number is of course maintained, nothing new, nothing added. Other things are associated with that. Please keep the conversation to what we're doing and don't have persons going all over the place. We really don't want to talk about anything but what we're talking about now. All right, I will pause and give you time to ask questions, but we have to be very disciplined if we're going to achieve the objectives today. All right, so another thing is that mitosis produces identical cells. I said that, and of course, it is very important for growth and development. Like I said, we really don't want to reach 1 year old and 2 year old, and things are a little different. And it ensures that there's continuity and variation. There is continuity, sorry. It ensures that there is continuity. What does that mean? It makes sure that the human, the species that has Homo sapien, it makes sure they are going to remain as Homo sapien, right? And the species that has crocodile, it makes sure that crocodile will always be here. And the species that has a mango tree, it ensures that a mango tree will be here. Not that next year we are going to have a new mango tree that is something different than the one. So you see there that mitosis is pretty important. Now, every time that you take a part of a plant, this is what it is now. So every time you take a part of a plant and you plant that plant and you get a new plant, it is mitosis at work through asexual reproduction. So for asexual reproduction to take place, mitosis is very important. Now, there's a formula to remember mitosis. Now, there's one thing I should have told you, is that mitosis pretty much produces two daughter cells. So at the end of mitosis, you are going to have two identical daughter cells. At the end of mitosis, you are going to have two identical daughter cells. Now, mitosis in itself can be represented as PMAT. And I'll just write it on the paper. I don't have a lot of paper, so I have to use space wisely. So it is PMAT. There we go. Now you can write these in the chat if you want to assist me. So PMAT, and the P there stands for, write it in the chat for me. The P stands for prophase. The N stands for metaphase. The A stands for telophase. Now that is pretty much yes, write them in the chat for me. That is pretty much what's mitosis. So prophase is going to be the first phase or the first step. Metaphase is going to be the second phase or the second step. Anaphase is going to be the third phase or the third step. And then telophase is going to be the final phase. Now, not to confuse you. Now, before mitosis takes place or mitosis begins, there is something that comes before, and that phase is called the interphase. Now, it is through interphase that the cell is prepared for mitosis. Now, after mitosis, there's another phase that is called cytokinesis. That is pretty much finalizing what happened in mitosis. Now, be mindful that interphase and cytokinesis, they are not a part of mitosis. Mitosis is only prophase, metaphase, anaphase, and telophase. Great. The cells, autosomal cells, are all the other cells in the body, except the sperms and the egg. So every other cells are autosomal cells, and they rely on mitosis. Great. Now we are going to get to the point of identifying the cells. And on your paper, you could be given a micrograph, and a micrograph is pretty much a picture that is taken under the microscope. You might have to identify it, or otherwise you might see it as an animation or so, just a drawing or something like that. Great. So what I'm going to be asking persons to just assist me with here, in interphase, what happens? An interphase comes before mitosis. What happens is that the cells start to prepare themselves, the chromosomes start to prepare themselves for the process of mitosis. So it's very important that we understand that. Now, what is happening? Like we said, the cell prepare itself. What is really happening with these cells? Tell me in the chat. So the chromosomes will, of course, become short and fat and visible, just to say the least, in preparing. Then what is going to happen in what we refer to as the start of mitosis, prophase 1, is that the chromosome, which is, of course, made up of two chromatids. So the chromosome is made up of two chromatids, and these become really visible. Now, the nucleus or the nucleolus is, of course, going to shrink and it will disappear. I don't know if that means anything to you, but the ring there that is around the nucleus, it is going to start shrinking and disappear. So you won't see that ring anymore. That's the easiest way to say it. All right. Now, we have what is called a centriole. The centriole. What will happen to the centriole now is that they are going to move. What's going to happen there is that they are going to move to opposite side of the cell. So if we are looking at a circle, they are going to move to the centriole is going to move to opposite side of the cell. All right, so you'll see these two big black dots at the center of the cell, while pretty much a chromatid in its pair forming that chromosome, they are still not so organized in the cell. Now, what we're going to have again is that spindle fibers, some thread-like looking thing, will somewhat be formed in the cell. And I do hope that you are following mitosis in your textbook as we talk. Now, the nuclear envelope, as we said earlier, it will just break and disappear. I think it's better to tell you that. That membrane around the nucleus, it will just break and disappear. So that tells us that we are pretty much at the start of mitosis. Now, the easy part of mitosis is that part we refer to as metaphase. And in metaphase, we are going to have the centrioles at the end. You see those little black dots there? They're at the end. And then we are going to have the spindle fibers coming off them. So we have some spindle fiber radiating, and we are now at the second phase. So the spindle fiber looks like this, like an onion somewhat type of thing. All right, great. And then we are going to have the chromatids now in metaphase all line up along the center. So there it is. They are all lined up along the center. That's metaphase. All right, it's lined up along the center. Now, it's very easy to remember metaphase because M, middle. M, middle. Metaphase, the middle. Now, what comes after metaphase? So we say that the chromatids, the chromosome, they are lined up pretty much along the center, right, of the spindle fiber or of the cell. Now, after metaphase, of course, we are going to go right into Anaphase. And anaphase is sort of something nice and easy to remember. Now we have metaphase here, so we're going to be drawing now anaphase. So anaphase pretty much, it has pretty much the centrioles, the two dots there just the same. And then what you're going to have, you still have the spindle fiber. But as anaphase suggests, away. Now what is moving away? What is moving away? Now the chromatids will pretty much pull themselves away from each other, but they are still identical and they are going to different sides of a cell. What does that mean? If we have a X, we are going to have something like a V going to one side and a V going to the other side. So just think about the X. And you just cut off the top part of the X and cut off the bottom part of the X. And then after you put them off, this one just say, okay, since we are separating, we're not staying together anymore. I am going over this side and you go over this side. You start your own family on this side and I will start my own family over here. And that is what is happening with here now with anaphase. They are moving away. So you might see the representation looking like a V. They are at different side. So you can see a space now where the queen can walk through the middle, right? In anaphase, you see a little space here where the queen could just walk through the middle and assess the guards to see if the guards are doing are properly clad and all of that. So here in metaphase, it's lined up right along the middle. And then in anaphase, what is happening now is that the chromatids, they are moving to separate sides of the cell. Are you seeing that? So you see the little thing here, they are moving away versus metaphase. So this is metaphase, and this is, of course, anaphase. Now, it's very easy because right after anaphase, we are going to have a telophase. And telophase is going to be that phase now where we are going to have some separation. All right. So the chromatids again will lengthen and unravel themselves because now guess what? They are forming their own family, right? So they want to get back to how they had looked originally. When they started out the cell division, now they are saying, hey, listen to me. I am just going to establish my thing just like my parent was. So now they start to unravel that chromosome, and then the chromosome pretty much lengthen. Now the other thing that will happen, remember they are forming back the family, so they are going to go back to what they were like. So telophase, they are going back to what they were like originally. So then the nuclear envelope, that membrane, will again be reformed around the nucleus there. And then another thing that is going to happen is that it's going to be evident that you have two groups of nuclei. Because remember that we had separate, and over here, hey, I'm going to form my family over here. And that happened in telophase. The last time they met in telophase. Sorry, I'm very sorry. The last time they met, so they met up in let us do this in perspective. So in metaphase, they met up and they decided what they were going to do. They decided that they were going to have one family over here and one family over here. Then in anaphase, they exact what the plan was. The plan was to have a family over here, so they started moving away. They're packing up their stuff and they're moving away. Some moving over here, some moving over here. Now, after anaphase, they get to telophase. And when they get to telophase, it's like they reach where they're going to establish their settlement. All right, so here now, they have to start unpack and start put back things like how it was originally. So originally, the chromosome, they are going to lengthen that chromosome, like I said before, they are going to lengthen the chromosome, and the chromosomes will, of course, unravel. The nuclear envelope will, of course, be formed around the chromosome, and of course, we are going to now have two nuclei. Another thing that will happen is that the nucleolus from each nucleus, there will be a new nucleolus from each nucleus. Now, that will result in two new daughter cells. But what does that look like? It's going to look like we have a box, you see, like we have a rectangle, and we have two new things in it. So all this thing, we have two new things in it, and this is what it is, because they are forming now. So if you look at the one at the bottom here, is that it's going to come like this. So now, in telophase, that is what is happening. You can see it's reforming itself. Pretty much it started as one, but now two new daughter cells will form. Now, what is going to happen after that is that, depending on which diagram you are using and which book, you might have a little cleavage. So you have to me drawing, I'm drawing a little cleavage here to indicate that it's going to start splitting down the middle here, so that it has one daughter cell over here, and one daughter cell over there. So now that process will, of course, be completed in cytokinesis. That process will be completed in cytokinesis. So now the process is completed. There's two new daughter cells, and that means that the cell would have been repaired or replaced. All right. So we have looked at some importance when we started out. Why is it that we should do mitosis? The function of mitosis. Now, mitosis is pretty much synonymous with asexual reproduction, but you're not supposed to go in depth with that for HSB. That is more a bio stuff. So remember, two identical daughter cells are formed as a result of mitosis, right? Remember, you know already that during metaphase, the chromosomes, they line up along the equator. They call it equator, and the middle of the spindle fiber. That is where they line up to make a decision. We are big man now, we are big woman, we have to go up on our own. What do you think? You think someone could have gone over some? So they have that conversation during metaphase. Now, during anaphase is when they decide that, okay, we have the conversation, let's get this thing done now. So they are now going away, right? They are going away. Now, in telophase is where now they say, okay, we'll reach where we are going, and now we have to set up at the camp, just like how the house was where we are coming from. And that is pretty much all that there is to mitosis. Remember, mitosis takes place in the autosomal cell, regular body cell. Then there is meiosis. And the thing about meiosis is that meiosis happens in two. Meiosis happens in two. So with mitosis, there is no variation in the offspring. All right, I think I should stop. Let me listen to what you're saying. Yes, you have somatic cell, autosomal cell. Yeah. God, me tired. Yeah. All right, that's fine. That's fine. All right, so that was easy. That is all that there is to mitosis for you at CSEC. Then we have meiosis. Meiosis is another type of cell division. So we talk about two different cell division. So if I wanted to make a new Mr. Wilson from mitosis, I'll just probably break off a piece of my ears or a piece of my nose or a piece of my eyelash, but just take up a piece of me and make a new of me. Mitosis only requires, when we're having reproduction by mitosis asexually, it's just one parent. All right. With meiosis, on the other hand, the first thing you need to know is that 4 daughter cells are formed. 4 daughter cells. So in mitosis, 2 daughter cells are formed. In meiosis, 4 daughter cells are formed. 4. The importance, or the major thing that stands out about meiosis, is that it brings about variation. It brings about differences. All right? The important thing about meiosis is that it brings about variation. All right? Now, meiosis is like twice mitosis. All right? But what I want to talk about, as it were, with meiosis, really, I think we should look at what we refer to as crossing over, which takes place in prophase 1. So you're going to have prophase 1, prophase 2. You're going to have prophase 1, prophase 2, and then just the same, just like how you have mitosis, all that part of mitosis, you're going to have 1 and 2 attached to them, as it were, with meiosis. The big deal about meiosis is that there is what we refer to as crossing over that takes place in prophase 1. Now, when I teach this, I just ask students to cross the finger like this, and what really happened is that a part of the genes here, are part of the chromatid here, will pretty much come off. The first joint will come off this finger, and it will go onto this finger. And this finger, the first joint will come off and go onto this finger. And that is pretty much what crossing over is. It's nothing bigger than that. So what you find happening now is that you have variation, because should this thing start mix up, you have variation taking place. All right, so you have variation taking place. Great. Now, another thing we need to know about meiosis. I'm not going to, the stages of meiosis is pretty much the same thing. And you're just going to see it come from one cell shooting out like that. Let me just find a past paper and show you what it looks like on the exam paper. Usually, when it is on the exam paper, is it on the 2020 paper? Just give me a second, guys. I have seen it on a paper, one of these papers I have here. All right, great. All right, so if you look at this, now you see the end product here, 4 daughter cell, you know that this is meiosis. So the end product there, 4 daughter cell. But then there is something else. It's not so visible on this because of the, all right, so pretty much the big thing here, the identifying factor here is that once you see 4 daughter cell, it's usually meiosis. Now, meiosis occurs, as we said, in the sex cell, and it happens there because the haploid number of chromosome needs to be passed on. What does haploid mean? It means half the number of chromosomes. So you and I, we have 46, but when we get together to have a child, it's 46 plus 46 is going to equal 1892. 92 chromosome. That would have been a different organism, not a human. So what happens is that meiosis divides this now into 2. So we will have 23 from mom, 23 from dad, and then the offspring will eventually have 46. But look at some other things that happen with meiosis. The offsprings are going to have some amount of variation. The offsprings will have some amount of variation. Now, the nucleus will, of course, have mixed chromosome. Why we say mixed? Because with meiosis, a lot of things are happening. A lot of things are happening with meiosis. First, you can think about the random fertilization. Just about any sperms could have fertilized that egg, right? And after any sperms could have fertilized that egg, then we have this crossing over taking place. So we have variation happening again. And then we have, we spoke about the haploid number. We have the 4 daughter cell. So a big thing about meiosis is that it ensures that there is variation, because you have this mix up like you're playing domino and you're shuffling the deck. You have this mix up taking place. So that is one of the big things about meiosis. Now, we want to ensure that we have a good grasp of that. I don't think they usually ask a lot of questions there. Let me tell you what was, well, we're going to do this later. match each label part. So we're going to look at haploid, diploid, DNA duplication, DNA duplicate there. All right, great. So yeah, so it's not much that they ask. All right. What I'm going to do now, I'm going to go through this one very quickly now. We are going to go through inheritance. And when we talk about inheritance, we're talking about how we acquire certain traits, how we acquired certain characteristics. Now, characteristic refers to those things that you can see. Is that fair to say? Some things that you can see are some things that can be found. Example, your height, your skin color, your eye color, all of those are pretty much considered to be characteristic. When we talk about inheritance, we are talking about something that you would have gotten from your parents or come down the family line, the family tree, that type of thing. So we talk about characteristic, and we pretty much say character. These are things that you can detect. They are either seen or found. Now, the characteristic, once we talk about characteristics, they are visible in the form of what we call phenotype. So phenotype refers to what you can see or what can be found. So for example, my phenotype for skin color is dark. My ear color phenotype is going to be black. If you talk about the phenotype or attachment of earlobe, I have detached earlobe, right? So you can see. If you talk about for the number of fingers I have 5 here. Some persons have 6. All right. So when we talk about phenotype, it's pretty much that trait that can be seen or it pretty much is found. All right, guys, please do not scare each other. Guys, please calm down. Guys, please calm down. All I am doing is to present the content. It doesn't mean that this is going to be on the exam. You will never know what is going to be on the exam until you get into the exam. So don't act as if it's all failure because you don't know what I am saying now. You never know. You might go to the exam and all that you know is what is on the paper. But what I'm doing, I'm just finishing up the broad topics as were presented by CXC. We were not able to finish it with you guys. We were able to finish them with our regular class. So don't get all jittery and start lick the chat with all sorts of talking, and then person start to gaze over the chat and they are not listening to what they could have learned. Person could have just grabbed a thing or two. Because what we know about the HSP paper, sometimes if you just have the knowledge and don't know it, when you look at the paper, you can match it based on a little thing where it hear or a little thing where it see. All right. So don't go confusing other. Don't go getting others scared. I suggest that you just hold your emotion a little bit. Don't have it flattering through the chat. All right. It's not the best thing. It's just going to be distraction. And if you are doing that, then it is sort of fear for me to just stop doing what I am doing. Because the Bible tells us no man can really serve two masters. So if I am talking here and the chat is all lit, how do we focus? What are we focusing on? The chat or the conversation? All right, so we have to make up our minds. There are some persons who want extra time. There are some persons who would have given up. But if you have given up, it is probably this is a good day to start for the next time around. All right, so don't just everybody's scared, not because other persons are not talking. They're afraid just like you. All right, so we understand, but we humbly beg that we don't leak that section and just start conversation that is not relevant. All right, so if we're going to be having that, then I'll have no option than to kick you off the live. All right, so we have to protect the others. Sorry, that is just how it is. That is the YouTube policy. What would that live be after the exam? Stop worrying about mitosis. Stop worrying about that and meiosis. What would the live be about? Yeah, stop panicking. Yeah, you'll be fine. Trust me, you'll be fine. I'm asking the classes after exam, what would it be for? Yeah, I can afford to take a break. What I'm going to do now is sort of not so difficult. Yeah, so persons are asking if I'm going to have a class after the exam. So I don't understand. So could you just bring me some clarity there? I'm really, really interested. Oh, you're asking if I'm going to be doing lives for paper one. Well, the only issue the only issue that we have is that we would have loved to have those papers on the screen. Now, in order for me to do that, my system can't carry the load of any software, so I was using StreamYard. For me to do that, I would have to find about 20 or 30 US dollars to pay StreamYard to have it for a month. You understand I don't generate revenue here. So I can give up my time, but have money, I don't have it to give. Now, if anybody can pay for that, then of course we're going to be here. All right, let me see what my assistant is saying here. Just let me read that. All right, great. So there, ADIPlus would have said it. Cross-stacking won't help. It will just provide more distraction and less time to get this thing done. You are going to have, you're going to get it. Don't worry yourself. All right, so we are going to Zoom. I heard that Zoom can stream live. I don't know. I'd have to check that. I don't know. One of the things with my policy is that I try not to have student crossing, right? Because I don't know anybody. And it would be unfair for me to hear that one of you come on my Zoom live and then next year your body is found somewhere. All right, so stop typing now and just let me answer the past paper stuff. We are going to try our best to meet you for the past papers for the question number one, or paper number one. We have 5 or 6 worked paper for you on the channel, but we are definitely going to try our best to meet you. All right, thank you very much. Let's continue. I need to have my pen for this one. All right, great. So we were talking about phenotype. So we said that phenotype are those things that you can see, those things you can detect, that type of a thing. All right, so we are going to ask you, please desist from using the chat now. If you are here to partake in what we're having, desist from using the chat. Unless you can't hear anything, you send us something. All right, please. Yeah, man, but we will treat you that. Remember, you can even send me a message after the live. Once you subscribe and click the notification bell, you can just click any video and send a message. I am always answering messages. All right, so we don't have to use this time to talk about those stuff. All right, we're sort of wasting our exam time now. All right, so I don't want anybody to become annoyed. So, we can use FaceTime. Let me write that down. But you could send that to me in the email too. Whatever you think I can use, just send it to me that I can just check it out and try it out. All right, great. All right, here we go. So, phenotype is that which you can see, or that which is found. Genotype refers to that genetic trait that would have caused the genotype. Usually, if we are doing an inheritance diagram like the pedigree chart or anything like that, the genotype is usually represented with letters. Just forgive me, I am now going back to the syllabus to make sure that I am following the syllabus in the delivery of a content. So we just looked at mitosis and meiosis. Now we are supposed to explain the importance. We did that for mitosis. It said the production of identical daughter cells have the same number deployed, the type of chromosome as the parent cell. Clones growth. All right, so I'll just copy something here that comes directly from the syllabus that is supposed to go along with mitosis. So I have been doing that since morning. So if they were supposed to ask you something on the exam, it is going to be something around this. It's there now. All right, great. So I am now going to go back to the syllabus. Describe process of meiosis. We did that. It's a definition of meiosis, movement, definition of meiosis, movement and separation of homologous chromosome, and the subsequent separation of chromatid, name of stages not required, diagram required. So you need to know the diagram. You need to be able to identify the picture changes, as it were, for the diagram. Now, explain the importance of meiosis. We did that importance of halving the chromosome number to haploid in formation of a gametes. I'm going to give you that as well. So the importance here, I'm going to give you that in the chat. Let me just put it on over there. All right, so here we go. So explain why genetic variation is important to living organisms. So we are supposed to, examples of variation, we're going to look at height, weight, gender, sex, blood type, tongue rolling, mention antibiotic resistant and bacteria. All right, just give me a bit. There's something I had prepared. This is going to be really, really useful to you. We have had some concerns about continuous and discontinuous variation, what really eye color is. This is a problem when I'm using these lot of papers because I have none. Just give me a second, I'll be with you in a bit. I did. All right, great. Probably you want to write this down. As it were, it says here that we are supposed to explain genetic variation, importance, and to living things. So first of all, there are some things that we need to know. And as you move forward in genetics, you need to know these. If you know these, the confusion that there exists when we are doing multiple choice will be removed. Now, there are some things that are inherited. They are caused by genetic inheritance and genetic inheritance only. So these, you could write things that are caused by genetic inheritance only. Type them in the chat for me. We're going to get over this very quickly. Stop worrying, trust me. All right, so things that are caused by genetic inheritance only are going to be blood group. Your blood group, whether your blood group is A, B, O, or AB, those are going to cause by genetic inheritance only. And these include, like I said, blood group, your ear color, that is all because of your genes, genetic inheritance only. Your eye color is caused by genetic inheritance only. Your sex, whether you're male or female, is going to cause by genetic inheritance only. Now, genetics will affect our genetics. This is caused by genetics, but it can be affected by environment. Your height. So your height is caused by genetics. However, it can be affected by environment. Now, your weight, how much you weigh, it is caused by genetics, but it is strongly influenced by environment. All right? So these are some salient points for us to remember while we are going through this whole concept of genetics. Now, I'm going to be going through, and I think I'm supposed to do some diagrams for you. Just let me check the syllabus. So explain variation, height. Just let me ask you, tell me in the chat, what is the importance of your height? Height bears any importance? I was looking at Daphne Skiffers or whatever her name is. I'm not too sure what her name is. Forgive me, if you know it, write it. I was looking at Skiffers, that fast girl from somewhere overseas there. And I was looking at Shelley and Fraser Price from Jamaica. They are both sprinters. However, Daphne, she is tall. Shellyan, she's short. Why is it that Shellyan is running faster than Daphne when Daphne has longer legs? So we decided yesterday, we were watching the race with them, and what we realized, though Daphne has longer legs, Daphne is lifting her legs higher than Shelley and Fraser. So by the time she land them on the ground, Shelley and Fraser would have gone. Now look at this. Usain Bolt is tall, but Usain Bolt has dominance in track and field. So what real advantage is gained by your height? Is there any importance? Yes, there's importance. There's importance. You understand that if you are tall, you have a better reach. And for games like basketballs and so on, that is an advantage, right? If you take, for example, the giraffe neck, it has a very tall reach. And then you will have smaller organisms that can just wheel themselves through small crevices and so on. And that could, of course, dictate whether they will live or whether they will die. All right? So another thing is that we need to look at height, your weight. You know that if you're overweight, how this could have pretty much affect blood pressure and diabetes. Your gender, does that affect, let's say gender or sex? These words are still words that are still not settled on. Your gender or your sex, how does this impact? What's the importance of this? It pretty much affect how you survive. Now, one thing you are going to know is that the whole physiological makeup of a man and a woman is a little different. The man tend to be stronger than the woman naturally. I think probably how the woman think is a little different, but does this help? These are things for us to look at. Those are things to be looked at. We're supposed to distinguish between genetic variation in the environment and include difference between continuous and discontinuous variation. And we're supposed to look at mutation and Down syndrome, and we're supposed to look at albinism. So we are going to be kicking on that now. Is there anything we want to ask before I move on? Anything we want to ask before I move on? I think no. All right, great. All right, so let us just start very quickly about continuous and discontinuous variation. And I'm going to make it very, very simple. With continuous variation, you could find varying degree you could have varying degree of that character, that feature that has been looked at. Let me give you an example. If you are looking, and you would have done this in your genetics lab, continuous variation. So you might have somebody that is a meter tall, 1.1, 1.2, 1.3, 1.4, 1.5, 1.6, 1.7. And you see there, we can get a nice gentle curve based on continuous variation. So continuous variation is usually represented with a line graph. Continuous variation is usually represented with a line graph because you can see that continuity. All right? See that continuity, that midpoint can be found. We're not at Punnett square yet. Not at Punnett Square, but we're going to get there. So what really, we spoke about continuous variation. Continuous variation is going to include, tell me some things in the chat, some things that we can include in continuous variation that will pretty much have a midpoint. Tell me in the chat some things that will pretty much have a midpoint. A midpoint is a distribution curve. So height is one of those things. Your height will help to determine your BMI. Height, growth height. But everybody keeps saying height. All right, weight, we're having something. Your weight will, of course, help you to plot a continuous variation curve. Size, weight, anything? What about skin color? The size of your feet, that, of course, is going to be continuous. Some persons have some big one like casket, and some have some little ones like pea grain. All right. So foot size, yes, you could look at that. The spans are the extent from one finger to another. Will differ from person to person. Intelligence, but I don't know how we fit that in. Some persons are going to be, I don't know if that is just I don't know how to put in intelligence. Now, let me look at what Amoy is saying. Usually, Amoy is usually backing me up from some end. So, continuous variation is a form of from one extreme to the next. Example, intelligence, while discontinuous variation is a clear-cut difference. All right, so we're going to go a little bit further with that. All right, so there we have some examples of continuous variation. There's a continuum. So I don't know that everybody understands, but I'm going to use this last analogy. You might see some persons on the road, and some persons are black like pot, and I love those ones. And there are some that are not so black like pot, and I also love those as well. And you have some that is not so, and you have that spectrum, right? Different shades of skin color, right? So that is where the continuity or the continuum comes in place. Then we have discontinuous variation. And with discontinuous variation, it speaks to is either something is or it is not. It does not have a mid-ground. It does not have a mid-ground, but it does not mean that it is really yes or no. The concept of yes or no can be used. So we could, when I teach it, the simple way to teach it is that it's either the light is on or the light is off. There's no mid-ground to say that the light is barely on. It's either the light is going to be on or off. I soon get to the examples. Guys. Just give me a second. So the discontinuous variation, it has no mid-ground. Guys, hold a bit with the chat. We soon get to that, and we want to ensure that, remember, when we are teaching and learning, we are using a sequence to learn, right? We're not going all over the place. We have to develop here, so go here, so go here, so go here, so we can't go up, like, down, and all over the place like that. That's going to be confusion. All right, so just follow my lead. I'm going to take your response. I really want you to talk, but just give me 1 little second. So the discontinuous variation, simply put, is either yes or no, right? It's either fear or no fear, and I'm really in the middle. It's either black or white. There's no ground for gray. It's like life. Life is black or white. There's no ground for gray. All right? It's red or white. There's no ground for pink. Probably I'm not using the best color stuff because a person might just be going astray. So let me start with me. So if you look at my ears right here, so you observe that this ears is not attached. This is detached earlobe. So it's either I'm going to have attached earlobe or detached earlobe. That is discontinuous variation. Attached earlobe or detached earlobe. Some persons ears itch on down here. So, well, my ear is big like an antenna, and then it have this little flap that if I'm missing the signal, it pick it up. I'm just kidding. Right. And then there are some persons who have widow's peak. So if you look right here, so on some person head, the ear come right out here, so that's a discontinuous variation. So it's either you have it or you don't have it. There are some persons who have hitchhiker's thumb. Poor me can't do it. You know them girl upon the highway where I stop the train. Hey, you're going my way? So I can't do it, but they are able to flip back this thumb and it bend back, some more. You see that bend back the thumb some more. Hitchhiker's thumb. Some persons have it and some persons have it. There's no middle ground. Nobody tell me about my maga, man. Oh, not so much, man. Me no maga. My body just dainty. Yeah, so if you look right there, so if you look right here, so you see one tendon here, so, and you see one next tendon here. So you can see the two of them. So the two tendon here. Now, some persons have two tendons there. Some persons have three. That is discontinuous variation. Now, some persons start telling us about eye color. Is either your eye is black, brown, pink, orange, or purple. So now that we know about discontinuous variation either being X or Y, not in between, give me the examples of discontinuous variation in the chat. Flow. Remember that the chat is being monitored. Remember, even when I'm not looking there, somebody is monitoring the chat. All right, great. So give me some example of discontinuous variation. Oh, my ears are like a bat. Yeah. Sometimes I use them to fly. I'm going to be honest with you. Sometimes I use them to fly. So tongue rolling. There are some persons who can roll a tongue and some persons who can't. Blood group. So it's either you're going to have A blood group, B blood group, AB blood group, or AO or BO blood group, or O blood group. But guess what? There's no mix. They are just something different. All right. We talk about blood type. I think what you call double joint is what we call hitchhiker's thumb. Hair length, that is something different. Ear length is going to be continuous variation. Some person have long hair, some person have short hair, that type of thing. Your sex is discontinuous. For the greater part, you're either man or woman. That is what it used to be. I don't know if you can be in between now. Forgive me. Careful. Careful. All right. Persons are still mixing it up. So just stop for me. Stop for me. This person must have just joined. Just stop for me. Lord of mercy. Am I? Please type in the chat examples of continuous variation. Am please type in the chat examples of continuous variation. Kenisha Johnson, please type in the chat examples of discontinuous variation. So Amoy will be typing examples of continuous variation, and Kenisha Johnson will be typing examples of discontinuous variation. All right, other persons, you can just wait on them to put the information there, because we want to ensure that we have that all good, lock and pot. All right, so we're looking at continuous variation. And then now I want the other persons who are not supposed to be doing the examples, I want you to give me now definition for continuous variation, and I want definition for discontinuous variation. So there you would have seen Amoy would have given you an example of continuous variation, intelligence, weight, and those are, there are some persons who are brighter than some. All right. And we are waiting on Johnson. So now we need some definition for both continuous and discontinuous variation. As you do that, I look at where we go next. We are going to go now to, okay, great. I need to just expound a little bit more on variation. I did tell you that variation speaks to differences, all right? So we want to look at variation and, of course, what really causes variation, what drives variation. It's very important that we understand what really drives variation. Continuous variation is where the characteristics show a continuous variation or show a continuum, all right? It's like a flight of step, you are going up, right? So that is what it is. Discontinuous variation is it is or it is not, all right? So there is just a cutoff point at somewhere. So we're having some nice things coming there in the chat. But what causes variation? And I'm going to be telling you what causes variation. Now variation is caused. The first thing we're going to look at is what we refer to as crossing over. That takes, well, it's not the first thing. All right. We could talk about variation. Well, probably it's the first thing. Variation takes place. We can think of one place where we have crossing over in prophase one. And prophase one is pretty much when we are doing meiosis. So we have variation taking place here. Remember, we cross the finger and things changing up and that type of thing. So my finger, this finger was black, this finger was white. And when we cross them over, this finger become black plus white up at the top. This one become white plus black at the top. And it mixes up the thing. Then another thing that causes variation is, of course, the division. While we're dividing during mitosis, there is some amount of, remember when we do that Punnett square stuff? There's just this random assortment of things. So variation coming there. So we have crossing over causing variation. We have the division during meiosis, and then we have the random fusion of the sperms and the egg. When a woman has sex, she really don't know which sperm is going to fertilize the egg. We have that random thing happening. Now, another thing that drives variation is, of course, environmental factors could, of course, influence variation. And as we're seeing in the chat, mutation, as we are going to be seeing with sickle cell anemia, mutation also drives variation. We talk about fertilization, we talk about the fusion of the male and of the egg and the sperm. So remember, the driving force behind variation is going to be all right, the driving force behind variation is, of course, going to be random fertilization, crossing over, division during meiosis, and all those are genetic stuff. And then we have mutation, and we're going to talk about the types of mutation. You want to jot them down when we get there. And of course, the varying environmental factors, as persons will talk about the nature-nurture controversy. All right, so that is a big thing to look at. The environment, if it's too, how does it affect you? That type of a thing when it comes to that. Now, another thing that we need to talk about is, let me check the syllabus here. Okay, we're supposed to talk about mutation. You really want to pay attention to this now, guys. We spoke about variation, continuous, discontinuous. We say what causes variation. You want to pay attention to this. And this is like a 2 marker so on your exam paper. But just jot it down for me. Jot it down for me. This variation. We're coming down nicely because I'm watching the time and I know exactly when we should finish to look at past paper and round it up. So I am looking at a lot of things. I'm looking at a lot of things. I don't know that we need a calculator for the exam, but yeah. So we're going to look at mutation. So my first question to you is, what is mutation? Mutation can drive variation. What is mutation? Just type that in the chat for me. So mutation is a random change in the genes. Mutation is something like, I don't understand that there. So mutation is a random change in the genes that could be caused by many things. It could cause by addition, deleting, or shift in the genes. All right. So mutation is a change in the genetic makeup or the genetic disposition or genetic. It's just a change. All right. And the change could be natural or could have been influenced by man-made factors. All right. Mutation is a change in the genes. Mutation is a random change in the genes. It's a sudden change in parts of the DNA. Mutation is a change in the order in our gene sequence, either due to mistake. Let me look at this one. This one is a little nice there. All right. So, like we say, mutation could be because of mistake when the DNA is being copied, or as a result of environmental factors such as UV light, cigarette smoking, and all of that. That's pretty much some of the driving force behind the mutation, and it will pretty much change. Now, what we want to look at for CXC Now that we have the definition for mutation, we want to look at gene mutation, and then we are going to look at chromosomal mutation. You need to write this down. Now, gene mutation is a spontaneous change in a part of the DNA. So it's this change in a part of the DNA, right? Which will of course alter the characteristics that pretty much control an organism. Now, an example of a gene mutation is going to be sickle cell anemia. So you want to talk about sickle cell anemia? Sickle cell anemia, Down syndrome child is a chromosomal mutation. All right, so just hold one bit, hold on me, just hold it a bit, hold it a bit, hold it a bit, hold it a bit, hold it a bit. No, BMI will not be on the exam because BMI fall under digestion. So it means that you just joined the class and you're not following what we've been saying. All right, please stop the typing now and let us just follow the command while we learn. Now, sickle cell anemia is a recessive condition. Now, the word sickle is pretty much from this farm tool that they use. It used to cut grass. It have this C-shaped type of thing. Now, when a person has sickle cell anemia, what happens is that the cell is somewhat off, or it takes this sickle shape. So it is not this full biconcave disc that we are used to. It's like half of that biconcave disc, right? Now, half of that biconcave disc, if you continue to do that, we have no option than to take you out of the room. It's half of this biconcave disc. Now, look at this. You had a big bucket carrying water, and then the bucket fell and it break off, and just a half of that bucket is left. Now, naturally, can that bucket still carry the 20 liter of water that it was carrying? Yes or no? Can the bucket still carry the 20 liter of water that it was carrying? Tell me in the chat. So you had a nice bucket there carrying, and then the bucket fell, it broke, half of the bucket is damaged. Will the bucket be able to carry the same 20 liter water, or it will carry less? No, it will definitely carry less. So when the red blood cell becomes sickle, it is no longer, it doesn't have this biconcave shape anymore, right? It doesn't have the depression in the middle. It doesn't have a depression in the middle anymore. What is going to happen is that it's going to look like this moon. Oh my, sorry. It's going to look more like this little moon. That is a sickle when you have sickle cell. So definitely the red blood cell will not be able to carry as much oxygen as it was carrying before. Once you have sickle cell, you cannot carry as much oxygen as you used to carry before. Now, with sickle cell anemia, I need to make this known. Anemia, there are many types of anemia. Whole heap of type. When you're seeing your period, you have anemia. All right, so there are many types of anemia, but let's look at sickle cell anemia. You can stop type now. Thanks, you were good just now. Now, sickle cell anemia is pretty much a type of anemia where there's alteration in the DNA coding for the hemoglobin. There's alteration in the DNA coding for the alteration. There's alteration in the DNA coding for hemoglobin. Understand, hemoglobin is very important in carrying that oxygen. Now, one part of the DNA is pretty much changed, and this is based on the wrong type of amino acid there being in the mix. So just remember that DNA mutation is responsible for sickle cell anemia. But this is where now we are going to come up and look at how a mutation, though it has debilitating effect depending on where you are, a mutation could very well have some good consequences, or could pretty much be good, could have a positive role. Now, once you're talking about sickle cell anemia, then this disease causes malaria, which is spread by which of the mosquito? That was a question for you. Malaria is spread by the Anopheles mosquito. Great. So once we talk about, no, not Aedes, Anopheles. Remember when we talk about Aedes, dengue, Ds, dengue, Ds, dengue, Ds. And if it is not the dengue and the Ds, then it's going to be the Anopheles and the malaria. All right? Always remember that, those persons who can't remember mosquito. All right? Great. So once we talk about sickle cell anemia, then the conversation must be had about, what did I say? Once we talk about sickle cell anemia, the conversation must be had about malaria. Why the conversation must be had about malaria? I'm going to share that with you. When you have sickle cell anemia, you will be killed by malaria. But malaria will kill you naturally. I don't have malaria. I don't have sickle cell, sorry. However, if I were supposed to contract malaria and it goes untreated, then I die. If you have sickle cell, it's even worse on you if you were supposed to contract malaria. But this is where the difference lies. If you have the trait for sickle cell, malaria doesn't affect you. That is what the mutation does. Now, some of you don't even know one thing that I am saying. So let me put it in writing. Let us think about doing this genetic cross. And we are going to define the alleles as sickle cell anemia. Let capital A be the normal person. So this person doesn't have malaria. Normal. This is a normal person. Now remember, not malaria. I keep mixing them up. Sorry. Sickle cell. We're talking about sickle cell. Malaria is a side effect. And we're going to define the other person, the recessive allele, for sickle cell as common A. Great. So the common A represents the person who will have sickle cell if they acquire it in the homozygous dominant state. We soon go through all of that. The person who is AA. So everybody is going to have at least a pair of allele. So this person with this 2 capital A, this person is normal. This person doesn't have sickle cell anemia. And this is what we call the homozygous state. We have the same 2 font size letter, 2 capital letter. I want to use letters now. This is homozygous. All right, so if it was 2 capital, it's homozygous. If it's 2 common, it is homozygous. Give me 1 second. However, this person, the second person here, this person would have sickle cell. But the third person here, with, no, we're not doing dominant such right now. We'll soon get there. Don't push too much word on them, please. All right, so the third person here with a capital A and a common A, that is the person who has the trait. And that person is called the carrier. Once you're talking about a carrier, they are going to have capital and common letter. It's a simple way to put it. All right? So the person with capital A and common A, that person is resistant based on the mutation, right? The DNA mutation for sickle cell. This person will not catch malaria. Are you with me? Anything for me to talk about? So once you're in the heterozygous state, which is, yes, heterozygous. Once you're heterozygous, it means that you have the trait. It means that you're a carrier. It means that you will not be affected by malaria, as it were, for sickle cell. However, if you don't have malaria, sickle cell, let me go again. However, if you don't have sickle cell, malaria will kill you. Once there's a possibility that you'll die. If you have sickle cell, there's a possibility, well, malaria can kill you. But if you only have the trait, you can walk around and you will be fine. As such, areas where there is prevalence of malaria, usually we have a lot of persons with the trait for sickle cell. So remember that sickle cell is a recessive case. It's a DNA mutation. We are going to come back to sickle cell again in this lesson when I'm going to teach you to do the genetic cross to determine if you had sickle cell and you were supposed to mate with another person, what the offspring is likely to be. So somebody helping me in the chat, and I think I like that. So capital A is going to be homozygous dominant. We sort of use it, but let's be careful. And common A is going to be the homozygous recessive. All right? And sickle cell anemia is a recessive trait. I just pray. I want to tell you this. A recessive trait does not mean that it is something bad. Having 10 fingers is a recessive trait. The dominant trait is 12 fingers. So a recessive trait does not mean that it is something necessarily bad. There are cases where it is bad. All right, great. So we looked at sickle cell anemia. If there is something else that I should touch on that, just type it in the chat for me. If a teacher or a student think that I missed out something, just type it in the chat for me now. You have privilege to type now. Please do so. Next thing we're going to be looking at now is chromosomal mutation. And with chromosomal mutation, the mutation takes place on chromosome 21. You heard what I said about Down syndrome? Where does the in a matter of fact, I've seen this past paper. I know your teacher don't teach you this, so you go and do a research now. I want you to type in the chat for me the 3 types of Down syndrome. You have to go to Google and get it. The 3 types of Down syndrome. Remember that Down syndrome that takes place on chromosome 21 is also called trisomy. The Down syndrome takes place on chromosome 21 is called trisomy. Now, I don't know that trisomy can be inherited, but Down syndrome, there's a type of Down syndrome that can be inherited. Let's get into Down syndrome. You know that persons who have Down syndrome, most of you would have watched 7 Heaven, or probably I watch it because I'm 1,000-year-old, so probably you guys want to say, yeah, old man, that's why you watch that. Then the show didn't go on again. All right? So we have translocation. Please write sentences that your classmates will follow. Don't just put a word. Remember that we have persons here with different learning needs. All right? So Amoy, I charge you with the responsibility. Type all 3 in one post and send it. All 3 types of Down syndrome or causes of Down syndrome, trisomy, mosaic, type all of these in one post and send it. Why I am doing that, I think in the book, or I think on the syllabus, we are looking at Down syndrome as trisomy occurring on chromosome 21. However, that is not the type of Down syndrome that is inherited. All right? I think it's a translocation that is inherited. All right? So you have trisomy 21, translocation, and mosaic. All right? Now I want you to tell me, I love you guys, Simon. I love you. If I had 2 hearts, I'd give you 1. All right. So I want you to tell me the type of Down syndrome that is inherited. The type of Down syndrome that is inherited. All right, great. So what happens here with Down syndrome, I'll tell you as we go along. When there's a diagram in your book, I think, that looks at Down syndrome, and it's the human chromosome 21. That is the chromosome that is usually affected by Down syndrome. Now, under normal cases, the haploid number of chromosome is passed on during meiosis, right? So you have even division. However, with a case of chromosomal mutation, you have what we call non-disjunction taking place. When non-disjunction takes place, what happens is that two of the chromosomes will end up over one place as what should happen over the place, over the other side when the division takes place, just does not happen. What happens here is as the process continues, an extra chromosome is added. So instead of you now having two, when the cell division is completed, you end up having three. You end up having three. All right. So usually when you see, if you are seeing a picture graph with that thing, I don't even remember what it is called. If you are seeing a picture graph on the paper, you are going to see three little dots beside 21, and you will actually see the chromosomes being numbered. Now, if you're seeing the chromosomes being numbered, and you see that three little dot, automatically you know that 21, three little dot, Down syndrome. All right. So remember that Down syndrome can be inherited, though it's also a condition that is caused by mutation. All right. So we in a crash course kind of way looked at Down syndrome, which is a chromosomal mutation, which is caused by the addition of a chromosome. And we also looked at sickle cell anemia, which is a type of mutation which affects the hemoglobin. Now, so those are some stuff that we were asked to address. I'm coming down, so we looked at mutation, we looked at Down syndrome. Okay, albinism. So I want you to look at albinism in your text for me. Now, albinism is also a type of mutation, pretty much like Down syndrome. Now, albinism is a case where you lose pigmentation in the skin, you lose pigmentation in the iris, and of course, the ear color also changes. That is albinism. Now, albinism is also another recessive condition. Now, we're going to be doing some genetic crosses that we're going to be looking at what is likely to happen if persons with somebody's condition happens to meet. 11:30, I am going to look after breakfast. I wish I had breakfast. Guys, remember you can leave at any time. All right. We are going to go as far as we can go, but we are also taking in consideration that persons are going to travel. And while persons are going to travel, we are thinking somewhere about 10:30 there, we should be able to get right into past papers that persons can leave for exam for probably about 12 or 11:30 there about. So we are cognizant of that fact. All right. So yes, when you want to go and look about your breakfast, yes, we have. I'll tell you something. We have quite a number of persons and persons have different needs, so we just have to treat with that. But we understand that some persons will go early. All right, not a bother. We really appreciate the fact that you're here. Great. All right, so we looked at that. Now we're supposed to explain the inheritance of a single pair of characteristics. This is what most persons want, and monohybrid inheritance. We should talk about DNA, RNA, stand for diribonucleic acid. And what I'm going to suggest to you, I don't know that this is a big one, but I suggest to you just get a small video on YouTube to look at DNA, RNA. We're not really going to get into that as such. There are some words that you need to know. And if you're using textbook, you need to know what inheritance means. You need to know what gene means, phenotype, genotype, homologous. They're all there in a table, some genetic terms. From the syllabus, you need to know DNA, which stands for diribonucleic acid. So you go, diribonucleic acid. And then you have RNA, which stands for ribonucleic acid. And then chromosomes, you need to know about chromosome. The definition for chromosomes that you have for that you have for primary school culture of protein is pretty much not sufficient. I think you need to understand that chromosomes are helix that are wrapped around a histone. I think you want that definition for CXC and not the little one that you had from grade 7. Now, alleles are alternate pairs of chromosome. What it means is one is tall, one is short. What if one gives the information for tall, one gives the information for short? One gives for fat, the other one gives the information for slim. And then we talk about dominant. The dominant trait is that trait that is visible in the phenotype if we are looking at inheritance of dominance. So once the dominant allele is expressed, any at all in the genotype, that is what will be visible in the, I don't know if that makes any sense to you. When I'm doing the cross, you'll understand. All right, so the other thing is that we're supposed to look at recessive. Or I should just ask you, somebody just give me the definition for dominant in the chat. The text here is saying that dominant in the heterozygous, I don't even want to read that, that is too long. All right, so recessive is usually that condition that you have to acquire in the homozygous recessive form. So if it's a recessive condition, you have to have 2 little letters in order to acquire it. Please look at the definition for all the words that I'm telling you now. And then there is homozygous. Homozygous, meaning that it's the same letter there, but get the definition. And of course, you can type the definition in the chat if you have them. DNA, RNA, chromosome, allele, dominant, recessive, homozygous, heterozygous, gene, genotype, phenotype. Those are definitions. They are usually 1 mark on the exam paper. You can look at that. Now, we are supposed to look at monohybrid inheritance. We should include albinism and, of course, sickle cell anemia. We should talk about tongue rolling. We should talk about sex link to include. So these are some things that we are going to do. Interestingly, I'm not seeing codominance determine the inheritance of sex in human. All right, include the role of the chromosome. All right, great. So we are going to do some activity now. So take out your book. This is the part that a lot of you are afraid of. It means that we are almost finished with genetics. So get your definitions that I gave you there. We are going to be doing some and I think another thing that I didn't say, but another thing we're supposed to look at is how mutation can drive evolution. All right, so we are going to be doing some inheritance of all sort of stuff today. And might I just say that inheritances are pretty much the same. What really makes the difference is the law that govern the type of inheritance. So usually when I'm teaching inheritance, I start with the simplest thing first. All right, so I'm not going to be checking the definition because a definition is just a definition. All right, Usually we start with the simplest thing first. So it is said that you have inheritance of sex. Some might say inheritance of sex, or some might say inheritance inheritance of sex or sex determination. So what that means, we're trying to find out what will happen the next time a woman, I think the woman who has the largest number of children is from Germany. So the next time she has a child, whether she had 1,000 boys before or 1,000 girls, what are the chances of her having a child that is whatever sex the next time around? Now, what we need to know is that the phenotype here for male and female, usually we define the alleles, right? So when we are doing a genetic cross, the first thing we define the alleles. So you can just write in here, yes, it's 50-50, but I'm going to do the entire thing that other students can see. I'm going to do all the genetic crosses, all right? So we define the allele. We are going to let XY be the male, and we are going to let XX be the female. And I think X and Y and XX, they are used for both alleles and chromosome. So there we define, that's the definition. Then, so the first thing we do is always to define. I hope you heard what I said, just do that. Number two, we are going to all right, guys, please calm down. Please calm down. Please calm down. This is serious business now. Serious business. But it's my area. Great. All right. So the first thing we're going to write is the phenotype. So the phenotype of mom and dad. So mom is, of course, going to be a female in most cases, or in all cases. And dad is going to be a male in most cases, or in all cases. All right. So the first thing we write there is the phenotype of the parents. That is what you can see. It's a found RC. Phenotype. All right. So after you define the concept, is to write the phenotype. All right. So the phenotype there being female and male. Then after we write the phenotype, we must write the genotype, the genetic makeup. And usually I don't think you do all of them writing here, and I think you just answer the question. I think them you do everything for you. So by a student who have to do all the writing. So this is the genotype, write the genotype there. So I'm going to fill them in. So you write the word to the left, genotype, and then you are going to write for female, it's going to be XX. And for male, it's going to be XY. All right? Can we just have a little humor? So when we teach genetics, usually the girls are saying that men are not really men. We are just mutation of women. And I'll tell you why they are saying that men are mutation of women, and we should not be boasting about we give any woman any ribs, because that is not what it is. You actually gave us life. We are real women. That is what the women are saying. This is just genetics trivia. This not have nothing to do with exam, but only too stiff. So if you look here, we have an X. So what the girls are saying in genetic cross, you are really a woman. You really should have been two X's. But because of mutation, I don't know, mutation take place. A part of the X break off right here, so and you become something looking like something called a man. So they are saying a man is just a hybrid like a mule, right? And then your clitoris just get enlarged and it become a penis. So we should be thanking the women that well, we should be apologizing to the woman for the whole idea that we said that we gave them a ribs, because we were actually women and we mutated to become man. I don't know if this is true. It's one of them foolish that I'm talking at genetics. It has nothing to do with the lesson. It is just one of them foolish that we talk when we're doing genetics. Let's continue the lesson. So that was the, of course, the genotype. Then after the genotype, we are going to write the gametes. We are going to write the gametes. And all we do when we are writing the gamete, instead of we write these so close, we space them out. So we space them out, X over there, so, and 1 over there, so, X here, so, and Y over here, so. That's all we did. We just space them out. When we're writing the gamete, all we do is just to space it out. If you are good like me, you just press print screen button every time I hold up this paper so that you don't have to wonder what I'm doing. Just print screen and have Microsoft Word, paste it in there, and you see what I'll do it. Now, after we have the gametes, the next thing we are going to have is random fertilization or segregation. Now, when we have segregation, that is where we are going to draw those line stuff. But I don't tell students to draw any line stuff, because when you start to do dihybrid inheritance or trihybrid inheritance, those lines can't help you. What we are doing now is looking at monohybrid inheritance, which is the inheritance of one type of trait. So we draw a Punnett square. A Punnett square is just a rectangular box. It's just a rectangular box like that. And then we draw two lines going horizontally in the box. We have two lines going horizontally in the box. And then we have two vertical lines. Vertical lines come from heaven to earth. Two vertical lines. And then the first cell at the corner here, we usually draw a diagonal line. Depending on what type of genetic diagram you are doing, sometimes you write male and female. Now, there are some symbols for male and female, like a male with a cross with a zero at the top and a woman with something like a triangle at the bottom. But if you can't remember that, just write the word male and write the word female. The word male and female is not really always applicable when we are doing a diagram. Now, what we're going to do, we are going to move the gametes from here and put them down into here so and up here so. So being that we have male at the top, we are going to put XY at the top right here. So we have XY, and I don't know for whatever reason the male is always on top. I don't know. All right. So usually we have the male on top. The male doesn't have to go on top. I don't know. It's some rude practice that somebody come up with. All right. And then we have the female at the bottom. So we have XX there. All right. Great. So now anywhere that we have an intersection, we are going to write the both letters. So X and X intercept right here. So we are going to have XX. All right. So where X and Y intercept, tell me in the chat, what are we going to have? Oh, which Punnett square? We are doing sex link. We are doing inheritance of sex. And might I say, a Punnett square can be drawn not only this way, but this is the easiest way to learn it. All right, so where X and Y intercept, we are going to have X and Y. Beautiful. What about where X and X intercept? What are we going to have? Where X and X intercept, we are going to have XX. Where X and Y intercept, we are going to have XY. So if you look at this, this is how it's populated. Wherever they intercept, that is what we are going to have. But it's important to note, these represent the genotype. If I were supposed to ask what is the chance of the parents having a boy or a girl, it's going to be 50-50. Because if you observe here, 2 girls, 2 boys. So the phenotype, girl, 2 girl, 1 boy, 1 boy. So we could represent that as a pretty much ratio. It would have been 2 to 2, but if it is ratio must be expressed in its simplest form. So what we have to do, we have to say 2 into 1, 1, 2 into 1, 1. So we end up with a ratio of the chance being a 1-to-1 chance. And if we are using percentage, it's going to be 2 out of 4 multiplied by 100 upon 1, which is of course going to give us 4 into itself 1 time. 4 into 100 goes 25 times. 2 times 25 is going to give us 50%. So there's always a 50% chance with the inheritance of sex that the next time a woman has a child, it's a 50% chance it's going to be a male, or a 50% chance it's going to be a female. You have this in your textbook, but if you don't have your textbook, I do hope that you can see it. So I'll just go from here. This is what it looks like. All right, great. So that was inheritance of sex. Remember, I told you that all the crosses are pretty much the same. It is just the rules that are different. All right. So now that we look at the inheritance of sex, we are going to look at the inheritance of dominance. Inheritance of dominance. Twin is something different, and it has nothing to do with this. Twin is something different. So we'll deal with that some other time. So we are going to look at the inheritance of dominance. Now, in order to look at the inheritance of dominance, you must know what the law says. The law says once the dominant allele is expressed in the genotype, the phenotype will be expressed as that dominant allele. What does that mean? Anna has the phone. All right, so let us look at this. I have a big head like a boom. And Anna has a small head like a small. Let me not use Anna. Anna's mother has a small head like a small. And we mated and we had Anna. Now, if big head is the dominant head, whether Anna acquired the big head as heterozygous or homozygous, meaning whether I'm acquire it with just one of the letter or two of the letter, he must have big head because big head is dominant. This is a dominant allele. It's a dominant trait. Detached earlobe is dominant to attached earlobe. So if I were supposed to have a child with a woman that has attached earlobe, the chances of the child having detached earlobe is really, really great. This is a dominant allele. This is called cleft chin, where the middle part of your chin here, it's sinking or have a dimple or whatever you call it, right? That's a dominant trait, a trait that will be passed on. But let us look at dominant allele. And I'm just going to pick something where we could talk about attached earlobe, right? So we're going to use attach, we're going to use the letter A. So we are going to say let A be the dominant allele. Let A be the dominant allele. And I have to shortcut when I write because it's going to take forever and I write like crab write walking in ink. So you write what I'm saying. Let A, capital A, be the dominant allele for attached earlobe, right? And then you are going to write let little a, that is common A, be the recessive allele. Be the recessive. So one is dominant, one is recessive. There we go. So now when we have the allele, we must have two parents. So we have two parents here. There is no carrier in this situation. So one, I have a detached earlobe, but I could have detached earlobe as heterozygous or homozygous. So let me do this and then you are going to do one. So I am going to be crossing a person who is homozygous dominant for earlobe. What that means, homozygous dominant, so parental phenotype, make sure you write that. And then I'm going to write the genotype. I'm teaching you, so follow the best practice. Don't follow me, I am naughty. All right, so the genotype here for the dominant earlobe is going to be 2 capital A. Now, my partner, she does not have detached earlobe. She has detached earlobe. So her earlobe, her thing is going to be 2 common A's. So her earlobe is detached. So mine is attached. That's my phenotype. My phenotype, I have detached earlobe. Sorry. My phenotype, I have parental phenotype, detached earlobe. My partner earlobe is attached. All right. Now the attached earlobe is a recessive condition. So remember to write that. Then after I started writing and making errors on the paper and crossing them out. So the parental phenotype is that AA is a parent with detached earlobe and common A is a parent with attached earlobe. Here we go. Next thing we are going to do is to write the gametes. And to write the gametes, all that we do is to separate separate the genotype. There we have it, the A's separate, and then I'm going to separate the common A's, and that's pretty much a gamete. I hope you can see there. Good. The next thing we are going to do, having got to the gamete stage, is now to do segregation. And to do segregation, all we are going to do, we are going to use our Punnett square to draw we are going to use our pencil to draw a rectangle. There's a rectangle. And then we are going to draw two horizontal lines, two vertical lines in the Punnett square. There we go. And then in the first cell, we are going to write a diagonal line. So there we have a diagonal line. It doesn't really matter who is male or female, so we don't need to put anything there. The next thing we are going to do, we are going to put A and A. We are going to put in the gametes now. A and A, capital A there. And then we are going to put common A down at the side there. Usually, the questions give a good idea as to how the layout should be. 9:59. So there we go. Now remember, wherever they intercept, that is where we are going to write both letters. So from what you're drawing, you have A, capital A, and what you're going to have is that all the offsprings will be heterozygous for the trait. All the offsprings will be heterozygous for the trait. Now the genotype here is capital A, common A. Now tell me in the chat, what will be the phenotype of the offsprings? What will be the phenotype of the offsprings? What will be the phenotype of the offsprings? Tell me in the chat. Phenotype. Phenotype. Phenotype has nothing to do with the letter. What will you see? What will be found? What will be represented? The letter is the genotype. The letters represent the genotype. The AAAA represents the genotype. The phenotype is what you are going to see. So, no, it can't be attached because the dominant trait is detached. The dominant trait is detached. So all the offsprings are going to have detached earlobe. All the offspring in this case will have detached earlobe. That brings us to the point that you have to pay attention to the question that is given. All right? I really don't have any time to go over it, but you could do other No, we don't use carrier and all that in this. No, we don't. You don't use the term carrier. All right, so let me get to what another one that CSEC want me to do. They say that I should look at, so I gave you sex link. No, I gave you, I gave you inheritance of sex, where it is again. All right, so I'm going to do albinism. I'm going to do that for you. All right, great. So all that I'm showing you, all that you need to know after you've learned the two first cross is just the rules. So here we're going to be looking at the next cross is albinism, and this is a recessive allele. So in order for you to have the disease, you must inherit your allele there in the homozygous recessive form. That means two common A's. It look like we're not so good here. Can I give you the questions and you do it and you give me the answer? Or you don't know what to do? What is a phenotype is what you can see. So the phenotype, when we talk about attached and detached earlobe, so the phenotype is either going to be attached or detached. In this case, the example I gave you just now, the answer is going to be detached earlobe. Guys, I see persons are making some requests, but everybody can make a request. Everybody have some limitations somewhere. So if we allow you to make your request, what is going to happen is that we're not going to be having a class because you are going to say E, somebody is going to say B, somebody is going to say C, somebody else is going to say D, and then at the end of the day, the donkey is going to ride us. So we have a plan, and we are going to go with the plan. We do hope that you can fit in, but we cannot answer the person's specific requests. We have over 1,000 persons in the room now, then we wouldn't be having a class if we're looking at that much. We understand that some persons are beyond here, but we have to make sure that we take care of what we had promised. All right. So I'm just going to shortcut this now because I show you how to do the definition up at the top. So I'm going to just shortcut this. Now, with the recessive condition, for you to have the disease with the recessive condition, you must acquire the disease with 2 small letters. Stop. So you must acquire the disease with 2 small letters. I did send persons last night to look at these videos on my channel, as the videos are already on my channel. I am going to be wrapping up this genetics thing at 10:15, at which point I will be looking at something different. All right? So don't worry yourself, everybody will be fine. So albinism, sickle cell, tongue rolling, good. We did sex already. So let me tell you what we have left to be completed here before persons start getting anxious. We just have specific objective 9, 10, and 11 left. Specific objective 9 said that we are supposed to explain the concept of genetic engineering. Now that you would have done somewhere, so I'm going to ask somebody just get what is genetic engineering and put it in the chat. So genetic engineering are changing the traits of an organism by inserting genetic material. Just let me just type it in, and everybody know about rice and all those modified stuff. So consider this done. Just let me put in genetic engineering, and then I'll come back. Then the other thing that we are supposed to do, we are supposed to discuss the advantages and disadvantages of genetic engineering, include the recombinant technology. Now, the recombinant technology would have been featured on a couple of papers, biology and HSB at the same time. I'm going to give you that insulin production. I would have said that to all my students. Make sure you know, oh, whoa, I forgot that is going to be on the paper. Just let me show you a diagram here. All right, so this is usually the diagram that is featured on the recombinant technology. Usually the diagram, this diagram here is usually featured on with recombinant technology, as it were, for CXC, with of course insulin production. All right, so if you have it, you can just summarize it. That's the diagram that is usually featured. All right, so let me just look at what else is there. That would have completed everything on genetics when we would have done that. All right, so just give me a second here. I'm still going to give you some time. Persons are yelling for 2017, but if you were here with us this morning, you would have heard me saying that you are supposed to go. We gave some papers that we liked this morning. Just give me a second. Let me try to find some. Which paper am I using here? All right, great. So we will soon do the 2017, 2020, and 2018 in short order. All right. So no need to worry. 2018, 2017, 2020. We're going to be doing these in short order. All right, great. these. We did these last night. Do that, and we gave some topic this morning. All right. So let us just continue and just finishing up. Let me just write out what I need to do, the genetic crosses that I need to do. I'm sorry I don't have a board. So when I did the SWAT this morning, you would have heard me saying that we don't have a board and we don't have the camera. So we are doing dominance, we are doing recessive, we are doing sex-linked, we are doing sex-linked. All right, so what I'm going to do, I did dominance already. I'm going to do the recessive now with the albinism and sickle cell. All right, those are recessive traits. So I'm going to be doing that now. But let me just tell you this. I am going to be jumping over the first part with the definition and writing out the gametes and all of that. I'm going to be jumping over that in the interest of time because I showed you how that is supposed to be done. So I'm just going to look at two parents now. One has sickle cell, and the other is a carrier for sickle cell. All right. So that is going to be AA, common A, two common A's. That is a sickle individual, and the person who is a carrier, that person has a capital A and a common A. All right. And these two persons are supposed to have children. So we are going to draw our Punnett square. And I'm asking you to do it now, you know. I'm going through it. That's why I'm not showing you on the screen. So we want to know what would be the chance of a child being sickle. So we have whose phone? Whose phone? Sure. Is that your phone? All right, great. So what is your answer? Tell me in the chat. What did you get when you do all that cross? Albino child, you got 50%? You couldn't get 50%. It couldn't be. Let me say it again. We are doing, all right. One of the parents is sickle. So if one of the parents is sickle, it is two common A's. That is going to be the genotype for the parent. And one of the other parents, is a carrier. That means that they have the trait. It is pretty much an heterozygous form. So they are going to have capital A, common A. We want to find out what is the chance of having an albino child. You should be getting 25% like the Sosa. All right? So just let me, it can't be 50%, must be 25%. Look at this. I skipped out the top part. See, the person who has albinism or the offspring is this person, right? These are carrier, normal carrier. They will have the trait. Malaria will not affect this person, but this person will be affected by malaria. All right? So it should be 25%. So I'm going to give you another one like how you get that one wrong. You are now crossing two persons. They are both carriers. So it means that two of them look at each other and them look nice and them thinks of them healthy and nice, but they are not healthy and nice. They have them nice. Something I walk around with sickle cell, something. No disrespect to anybody, all right? Great. So what is going to be the result for this? I want you to write the genotype in the chat when you are finished. What is going to be the genotype for this? So after inheritance of recessiveness that we are doing now, that has to do with albinism and sickle cell, we are going to do sex-linked inheritance, and that's going to be the end of inheritance, end of continuity and variation, as it were. The correct number for the one we did a while ago, I showed. It is 25%. The chance here for the one we did with the sickle parent and the carrier parent, the chance of having an albino child is going to be 25%. Only one person. Now, for the one we're doing now, where both parents are carriers, we are looking at what the offspring is going to be. Please write everything at the same time. Well, I'm going to do this time. All right. Jardine, you're looking good. Jardine has the answer. So the answer there should have been capital A, which is homozygous dominant. Then we are going to have 2 carriers, and 1 person will, of course, have albinism, will be an albino. 1 person there, the 2 common A's, that person will have the albino thing. So that graph, that Punnett square, should look like this one here. And I hope you can see. So the one at the bottom here, good. All right, so the chance of a person being albino when we have 2 carriers, a 25% chance. Now, we are now going to be looking at the hardest. Well, it's not the hardest. I think codominance might be the hardest for most students. And I don't see codominance on your syllabus. Instead, I see sex-linked inheritance. With sex-linked inheritance, it's very important that when we communicate, we say male and female and whether they have the disease or not. Now, with sex-linked inheritance or X-linked inheritance, what happens is that the trait is carried on the X chromosome only. The trait is carried on the X chromosome only. And those diseases that we are looking at is going to be like color blindness and hemophilia. Now, hemophilia is what we refer to as bleeding disease. When you get a cut, you have to take medication for it to stop. Or when you are color blindness, you are not able to see some spectrum of light. Now, I'm going to just do one for you, then up on into the past papers. As persons are getting a little jittery, it's time to move to the exam, and I want to give everybody an opportunity. So we're going to be doing the last cross here, which is sex-linked inheritance. So we have a female, and when you have this, these are normal. Remember, the Y will not carry the trait. It means that a man is going to either have the trait or is either going to have the disease or don't have the disease. The woman can either be carrier, normal, or has the disease. So the woman has 3 stands that she can take. The man can only take 2. It's either I have the disease or I don't have the disease. So now we are going to put on the trait for hemophilia. And I'm just going to use H, capital H, common H. And for the man, I'm going to use a capital H. And this suggests that this man is normal, being that we have the capital H there. But the woman is a carrier. So now we are going to cross these 2 individuals, and we are going to determine what the offspring is going to be. Now, in this case, it's very important for us to write male here and female here. So we have male and female. We're going to write the male at the top, which is going to be X with a capital H and Y. So this pretty much represents the male. So the male is in. Now we're going to write in the female, which is going to be 2X with a capital H and another X with a common H. Now, if you observe here, we are using the sex of the parents. That is very, very important for this type of genetic cross. The next thing we are going to do, wherever the letters intercept, we are going to write them both in, and we are going to make sure that we carry that trait, that we are sure what the offspring is going to be. So there we would have written in the first offspring. The next thing we are going to do is to just continue and populate this entire table. And if you observe here, while we populate the table, we are getting both male and female, as the inheritance is going to be determined based on your sex. So in this case, if you observe, the offsprings are male and female. Some will have the disease and some will not. So I am going to now be numbering the cells, and you are going to tell me those persons who have the disease and those persons who do not. So I'll number the cells 1, 2, 3, 4, and you are going to look at the screen and you are going to try to name. Now, to name them, you must say female carrier or female hemophilic or male hemophilic or the person doesn't have the disease. So there we have it. So we have 1, 2, 3, 4. You are going to tell me what is 1, what is 2, what is 3, and of course, what is 4. This is going to take us to the end of genetics for this morning. I do hope that all will be well for you. We are going to up one into some papers, and those persons who can manage, you are going to help us to go through the paper. Anna is talking to me on the phone. Any answer? That was a kill? All right, so I'm going to help you. So we are going to have one normal female, and I'm going to draw arrow. One normal female. We are going to have one carrier female. So there, oh, you're lost. I'm sorry. I'm so sorry. So we have one normal female. There it is, and I draw use some arrow to show where it is coming from. And then we are going to have one normal male. And I think somebody has it there. And we are going to have one hemophilic male. If we have time, I will go through this at the end, but that's pretty much what the result is saying. If you observe, the way we communicate the answer here is a little different. All right, so one normal. No. Oh, you're giving the number. So 1 is going to be normal. 2 is going to be normal. Yes. 3 is going to be a carrier female. And 4 is going to be a hemophilic male. That was beautiful. Wonderful. Great. Give yourself a stretch now. We are 4 minutes over time. We are now going to try our best to see how we can answer these past papers. And then you are going to run on to exam while I will run onto the road to get some things done. Have a look at the paper sometime today. And then probably we post the paper sometime tonight. Probably by about 9 o'clock, 8 o'clock thereabout. We post the paper. Now, as to whether or not we are going to be having marathons for, what is it called? Multiple choice paper, whichever way we are going to try. We are now going to be looking at past paper 2017, which seems to be in high demand. We're not going to be doing the first question because that is on digestion, and digestion is on here stuff. Digestion is a life process, don't it? The syllabus, they are different. Let's let me check that. Digestion section B, great. Yeah, digestion in section B. So we're not doing that. We are not doing number 2 because number 2 is not on the exam. That's circulation. But we're going to start at number 3. And number 3 is pretty much the skeletal system. All right, great. So I am going to be showing you this thing, and you are going to help me to answer as we bring the curtain down, and I wish you all the best for your exam. Figure 2 shows a diagram of a muscle, bone, and joint of the upper arm. Remember, I told you that you need to know this diagram for your exam. All right, remember I told you that. Now, diagram showing muscle, bones, and joint of upper arm. Now, which letter indicates the tricep? That's what I like about HSB. You can just guess your way around. So you are going to look at this now because we did this last night or some night. Which letter represents the tricep? I hope you can see which letter represents the tricep. Can you see? All right, so which letter represents the tricep? So Anushka send the paper to everybody via Instagram. Yes, so Mr. Wilson know everything. So please follow on the paper. Anushka, you sent it, right? All right. I see a whole heap of wrong answer, and I see a whole heap of right answer. Some person can't see the lines that are drawn. This is the tricep muscle. The one at the back, which has 3 tendons, this is the tricep muscle. This one around here, so this is the tricep muscle. So for the persons who selected F, your answer is correct. I know that some persons can't see the line. The persons who selected F, that is your answer. Okay, great. Anushka is a nice person. Now, which letter indicates the origin and insertion of a tricep muscle? Everybody who come to my class know that I am highly animated. When we say that which part you come from not change. If you come from Timbuktu or come from which part, you have to duck when you see the plane, it can't change. That is the point of origin. That bone seldomly move or moves none at all. The part that moves is the point of insertion. Remember, I said that and I did the same thing. So now we are going to look at this diagram. Let me see what I'm going to do now. What is the point of insertion here? Remember, we're looking at what we're looking at. The upper arm, we are looking at the scapula and the. Remember, we did this. So the point of insertion, we're looking at the scapula and the radius. How persons get D? The scapula and the radius we are looking at. If we are looking, what are we looking at? The point of insertion or origin. We are looking at the point of insertion. What is the point of insertion? I can't even see this thing good. So the point of insertion. Yeah. No man, insertion. Insertion are gonna be down a C, down here, so we connect. Here so, or the insertion could be here, the back here, this tendon here, or up at the top. So I think it has more than one answers. All right, so we are going with C, and I think G could be used as well because that is moving a lot. I think C or G. And then the point of insertion is where the muscle connect to the scapula. Point of insertion is where the muscle connect to the scapula here. So I think we could use E or A, don't it? All right, you can correct me. It's going to be E or A. So it's going to be G or C for insertion and A or E for origin. All right, so we move on. Identify the type of joint at D. The type of joint at D. Type of joint at D. Type of joint at D. I don't see the thing. You can't see the paper. But Anushka sent it to everyone. Okay, I am just looking at, let us not get this whole idea that the point of insertion and origin only has to do with the bicep, only has to do with that. So I gave you 2 ways in which you could look at it. But it's usually, I am even looking in the book right now to make sure that we are on par with what I said just now. So don't kill yourself too seriously. I gave you 2 options, but I'm making sure. I am that type of person who want to make sure. So insertion is correct. I can't find this gap. Yes, I am so correct. What I said is true. I am going to show you the diagram in the book and I'm going to point it out. If you look here, it is looking at both tendons here and here as point of insertion. Point of insertion here and here, depending on whether you're looking at the bicep or you're looking at the tricep. Then the point of origin, it also goes to the top here, and it shows you that for the bicep and the tricep, the point of origin. So what we said just now was indeed correct and keeping with the textbook. All right, so let us move on. Identify the point labeled D. That's, of course, a hinge joint. I don't know why we're having so much confusion. It's May, June 2017 paper that we're doing. May, June 2017. Then the last part now, it says state the type of movement that joint D allows. State the type of movement that joint D allows. State the type of movement that joint D allows. Okay, want to see the paper again? All right, so joint D, state the type of movement that is allowed. Back and forth, movement in one plane, freedom of movement in one plane. So it allows movement only in one plane. All right, great. So we move on to the other question, and it says, Diana's personal trainer asked her to complete 20 bicep curls. The exercise requires her to flex and extend her arm. Discuss how the bicep and tricep muscle work together to achieve this task. So now they are asking you to explain how antagonistic muscles work. So you are going to tell us how antagonistic muscles work. How is she going to do this? Remember antagonistic muscles, how they work? Type that in the chat for me. And we are burning, we are going like wildfire. All right, just tell me how that works. To work antagonistically, the bicep contract while the tricep relax. Amar, you have to include in that how much a question value. The question value 4 marks. You wouldn't get 4 marks for that. So you have to tell us that when one contract, one relax. You have to tell us about the muscle shortening, the muscle contracting. You have to tell us about the muscle becoming bulge. All that must be a part of the thing for you to earn that 4 marks. Antagonistic meaning when one muscle contract, the other muscle relax. That is not enough to answer the question. You have to explain exactly what is happening there. Action of a muscle. I don't understand why persons don't know how to answer this question. And we told you that this question is something that you need to know. Do you have that? So I showed you before, but I'll show you again. So now the hand is extended. In order for the hand to flex, it comes up like this. When it comes up like this, what happens is that the bicep muscle contracts and become bulge, or it becomes fat. You said it last night. While tricep muscle will relax. So the muscle under the bottom here will relax. So the bicep, once the arm is flexed, the bicep contracts and become bulge. The tricep muscle will relax and pretty much lengthen. And the reverse is true. All right? That is what they're asking you to explain here. How does this happen? All right? And you must include in it that it's an antagonistic action, or they are antagonistic muscle, which means that they work against each other. Next question. Diana is exercising to tone her muscles. It's not about what time I'm finishing. It's how long it's going to take you to get to the exam. So when you have to leave for exam, leave for exam. Don't stay here and be late for your exam. All right? I have 3 papers to do, and we're going to try to do that, and then we can leave. All right? Preferably I want to leave here by about 11:30 there about. All right? Great. So Diana is exercising to tone her muscles, state what is meant by muscle tone, and suggest how it could benefit Diana's joint. So I talk about muscle toning. What does muscle toning mean? So Amma is all ready for the exam. What does muscle toning mean? To tone your muscle, what does it mean? I did this in some live 2 or 3 days ago. Anna, what's wrong? Come here. Come here. Come here. Am I calling? Come here. What I'm like? All right, so muscle tone is pretty much just tension on the muscle. All right, so when we talk about muscle tone, it is pretty much having tension on the muscle. Right, that is so correct. Now, the next question is tension on the muscle when it is relaxed. The next question is, how does this muscle tone help the joint? How does the muscle tone help the joint? Faster you go, the more we can cover. How does muscle tone help the joint? What's the benefit of a muscle tone to the joint? We know that the skeletal system is going to be on the exam, and we are almost certain that this is the question that is going to be there. We are almost certain. So we're looking at how muscle tone helps the joint. So if there's a tone on the joint, what is going to happen is that it's first thing it's going to help to keep the joint in its correct position. So when you are supposed to move off, it's like you're almost ready to move. You want to think about any extra muscle to move before you get moving. It's like you're more alert. So a lot of persons are saying it maintains that balance. It helps to keep, that is true. It helps you to keep that balance, that proper posture. And of course, it gets that joint in that position, ready to go. Balance, good upright posture, and keep it in an active state. All right. So that is what it does to joint. It helps to keep the joint pretty much in the correct state in order to keep that good posture and to put you in that alert state to go. Keep that shape. All right. Good. Our next question is going to be Diana needs to lift a 50 kg weight. Her trainer instruct her to maintain proper posture. Advise Diana on the correct way to lift the 50 pounds. So here we go. That's a question. Now you're going to tell Diana how to lift the weight. I'm sure that we did this in one of the lives. Yes. Good morning. Good morning. All right. Yeah. All right. All right, so she's supposed to bend at her knee and lift with her. All right, so she's going to lift with using those muscles in the leg, bending the knee, keeping the back straight, and coming upright. For those persons who don't know, let us just show you the diagram very quickly. This is the diagram and how she should lift, the one with the knee down. That is how the lifting should be done. All right, great. Let's move along, and we are using this textbook to assist with the illustration. That's not our work, and we claims no right to it. Suggest what would happen if Diana does not maintain proper posture. So if Diana doesn't have proper posture, what is likely to happen? If Diana does not have proper posture, what is likely to happen? So if you will end up having pain and you will end up having hollow back, round shoulder, this is proper posture here. So if you don't maintain proper posture, your body, we did say that in a live, that you are going to be twist, you won't twist, round shoulder, hollow back. I spoke about that. And of course, you will have pain sometime because the body is just not it's not balanced. All right. So back pain is a common feature when a person has poor posture and of course poor digestion of the food that we eat. Another question we're going to be looking at is question number 4. Question number 4 speaks to homeostasis. Homeostasis is necessary for the proper functioning of the body. It includes negative feedback and positive feedback mechanism. We have been doing that for quite some time now. Define the term homeostasis. Let me have the definition for homeostasis, please. Definition for homeostasis. Definition for homeostasis, please. Round shoulder, back pain, hollow back, all of those are things, signs, symptoms. If you have not yet and you like what we do here, please be reminded to like, share, subscribe. And remember, when you subscribe, click that notification bell and select all. If you see how many times we come on and have to do over this thing, if you have not selected all, you are going to be missing a lot. All right, homeostasis is the maintenance of a constant internal environment. That is what it is. I'm not going to look for anything else. Name 2 other organs in the body that is involved in homeostasis and state one product of each organ that each organ excrete. Write your answer in the following table. So here it is. You are going to talk about organs that assist with maintaining that internal condition. Look here at this, and you are going to write it in. You can stop right about homeostasis now. Oh, well, continue, do your stuff. It's your thing. All right? So identify 2 organs. I'm sure we did that last night on one of the past paper that we did yesterday. So do that for me. We are watching the time. We still have enough time for some persons. Remember, the exam starts at 1. So you don't want to be here and left behind. You want to be at the exam on time. So we're looking, it says here, excretory product. We're looking at the kidney, and the kidney is going to get away urine. And of course, we have the skin getting away sweat. We are going to have, you can't just write skin, lungs, and kidney. Say what they are doing. And the lungs getting rid of carbon dioxide and some amount of water. We have sweat coming from the skin. I think we are ready for this question. Let's move right along to the other question. Let us see what the question is asking. It says, Kenrick has the first meal. This is a nice one. I have done this a million times. Kenrick has the first meal of the day at 7 a.m. It consists of toast with peanut butter and a glass of juice and some fruits. He works through the day and does not eat again until 4 p.m. After digestion of the toast, glucose enters the bloodstream. Explain the role of the hormone in causing the glucose to enter the cells of Kerrick's body? What is the role? Explain the role of hormone. So after digestion of a toast, so the toast is broken down, you are now going to, glucose enters the bloodstream. Glucose enters the bloodstream. You are going to explain the role of a hormone in causing the glucose to enter the bloodstream. Let's have that. What's the role of the hormone? The role, what role the hormone plays? Not what is the name of the hormone, it says what is the role, what the hormone does. What's the role of a hormone? Let me read it again. After digestion of a toast, glucose enters the bloodstream. All right, so let us look at what happens after digestion. After digestion, some of the glucose is converted by insulin. Okay, the insulin tells cells throughout the body to take in glucose from your bloodstream. As the glucose move into the blood cells, your body glucose level will not go down. Sorry, your blood glucose. Just let me read that again. I think I like what is here. This is where we should be going with this answer. It values 2 marks, but it takes something like this. So the insulin tells the cells throughout the body to take in glucose from your bloodstream. As the glucose moves into your cells, your blood glucose level go down. So that is what is going to happen. And if the blood glucose level continues to be too high, then the liver, with the aid of insulin, will of course convert that extra glucose into glycogen, and it will of course be stored in your muscle, liver cells, some converted to fat, stored under the skin and around organs. Next question. Around 4 p.m., Kenrick begins to feel restless and suspect that it is as a result of not eating. Upon checking his blood glucose level, he realizes that it is very low. Explain what would take place in his body to rise the blood glucose level. We have been doing this. What is going to happen now? Now that the blood glucose level is low, what is going to happen inside the body? Remember, we had some stores from this morning that the cells didn't use. What is going to happen now? This morning we had converted some of that glucose into glycogen. What is likely to happen now that he's restless, he really needs some energy? What is going to happen? To say that pancreas will release glycogen, that is not adequate. What will convert glycogen to glucose? If you observe here, we have interest in the name of these hormones. I have been saying this for some time now. All right, Heinz, I'm seeing you. So glucagon will convert glycogen back to glucose and make it available for use. Remember, glucagon converts glycogen into glucose. You need to remember that. Let's move to the other question. As a result of being so hungry, Kenrick eats a very high carbohydrate and fat meal, and soon falls asleep. Suggest what would happen to the excess glucose formed after the digestion of the carbohydrate. What is going to happen to the excess glucose? Fell asleep, still have some excess glucose. What is going to happen to it? All right, so the excess will be converted into, again, glycogen. And if there's still excess, it's going to be converted to fat stored under the skin and around organs. But you would have recalled me telling you that you can't go to this exam without knowing about diabetes and hypertension. This question is to look at those. And the question reads thus. The effect of lifestyle-related disease on human body can be significantly reduced or managed by diet and exercise. Explain how diet and exercise can reduce the risk of one lifestyle-related disease. Now, if it says that one lifestyle-related disease, you have to name the disease and then you tell how diet and exercise could, of course, help with this. So let me hear your disease and how diet and exercise can help with it. 7B on the syllabus, it takes asthma. Asthma is not an infectious disease. But this comes with something to explain the difference between infectious disease and communicable disease. Now it's an infectious disease, and okay, now I understand. Thank you very much, Matt Milan. It was nice having you. I'm waiting for that answer. Come, Amoy, why are you giving me those skimpy wimpy answers? Campbell, I like what you have there. I was expecting persons to whooze out things around hypertension and diabetes, but I'm not seeing that. So I take it that you have not really done that groundwork as I'd asked you to do. Exercise lower blood glucose level and boost your body's sensitivity to insulin. Let me just say that. Okay. The insulin, countering insulin resistance. Many studies underscore these and other benefits of exercise. That sounds pretty good. But what disease are you looking at, Drummond? I don't know what disease. It did say with relation to one disease. So you have to name the disease and say exactly what you're talking. You have to name a disease there. You can't just give all that fancy definition, which sounds really, really good. But guess what? We don't know which disease you are talking about. So you have to get that under wraps. All right, great. So we are almost at the end of this paper. We are at question number 6, and question number 6 looks at bacterium often found in raw sewage. And this is the last part of the syllabus, section E. Bacteria often found in raw sewage is vibrio-cholera. State the disease the bacterium causes and give 2 symptoms of the disease. So the bacteria found in raw sewage is vibrio-cholera. You are supposed to state the disease that this bacterium causes and give 2 symptoms of the disease. Cholera, 2 symptoms of the disease. You see how the paper easy, you could just listen vibrio-cholera, then in a must cholera, brother. So what are some of the symptoms of cholera? I think we did that last night as well. What are some of the symptoms? Remember the difference between symptoms and signs? We did that last night. So cholera is always going to be featured when it comes to sewage and water contamination. All right. It will cause fever and vomiting. We did say that it is going to cause gastroenteritis. That is inflammation of the intestine. You will have stomach pain. No, ringworm is something different. Diarrhea as a result of the gastroenteritis. I think you did well there. Now, I did ask you to look at the construction of pit latrine. Now, this question is around pit latrine. Describe how the drop latrine shown in, I soon show you, figure that, operates, and recommend a suitable alternative small scale sewage drop disposal solution. Justify your recommendation. So let me just show you this on the screen. Look at it and give me your answer. Yes, dehydration is big there, Amoy. I'd forgotten that one. So answer that for me. Let me read it again. Describe how the drop latrine system in figure 3 operates and recommend a suitable alternative system, suitable alternative small scale sewage disposal solution. And then you are supposed to justify your answer. Water carriage is nice, but tell us about that. Yes, water carriage is an alternative method that is spot on, but tell us, how does the drop latrine system works? Sure. When I All right, what do we have? All right, so a septic tank is an alternative, water carriage is an alternative. Repeat the question. Great. Let me repeat that. Remember, we have 2 more papers. Amoy has service on her phone. Some persons have service on their phone and they are traveling with Mr. Wilson live. Hey, hi to all the persons on the bus. Let me say hi to you. This is CSEC Biology TCP. We are on YouTube and we're assisting these students in getting the 1 for biology and HSB. You want to subscribe as well. All right, great. So let us read the question again. Describe how the disposal, describe how the drop latrine are shown in figure 3, or we call it pit toilet. All right, operates and recommend a suitable alternative small scale sewage disposal solution. And you're supposed to justify. I see a lot of persons giving the alternative, but as to how the pit toilet work, you can't tell me. You guys don't have too much flush toilet down here. If you don't come from country, you must know it work. All right. So describe how the drop latrine. Okay, great. Somebody put the answer there. Sir, drop latrine mean it's raw sewage going into the soil, may cause, not too sure what happened there. They lift the lid. All right. Let me look at this one. I hope nobody's having lunch now. They lift the lid, then they drop the cases inside, then throw sand or lime over it. So every time they do that, they throw sand or lime over it. Drop latrine is used in case with little water, a hole is dug and the ground in the ground at least 3 meters deep with a seat over the hole. It does not use water. But you did not tell us about the that casing. Remember, I'm going to show you another drop latrine. Just give me a minute. There's another drop latrine on a paper, and this one is it's like a water carriage system. Don't even remember which paper it is on, but I've seen it on one of these papers. Which paper? Just let me show you something here. Look at this fancy one. With vent pipe and all of that. This is a different one. Which paper is this? This is on the 2018 paper. All right, so how does it work? Drop latching. Well, it's pit latching is when the human feces. Okay. Simply put, it has a lid. A hole is dug into the ground, and of course, it is so built that no rodent can enter. It is about 3-4 meter deep thereabouts. So the depth, when you explain this, the depth of the pit latrine is very, very important. There is no water to the pit latrine. So what you do your droppings there, and having done the droppings, then you are going to, I don't know that you just leave, I don't know that you just choose sand or anything like that on it. But what I'm getting here from you is that you need to do one last reading on pit latrine before you get into the exam, because you really don't know much about the pit latrine based on what I'm seeing here. All right, you need to know about the construct, where it is constructed, how close it should be constructed to water, type of soil type that you should use, how close it should be constructed to the house, that type of thing that you should know. So what I'm seeing here on pit latrine, it says that your knowledge on pit latrine is really, really weak. You have until 1 o'clock to get this right, and it's just a small portion to read. So I hope that you would have walked with your book to read on the pit latrine. All right? So you have pit latrine, you have different type of latrine. I am not satisfied with what I have seen here for the answer here, so we have to do some more. All right? Because it says we are supposed to. You are supposed to describe how the latrine work. Right? And you can't tell me how the latrine work. You have a little bit of knowledge on it. I'm not so comfortable with that. All right. So please, just fold that page in your book. You have to read it. We're going fast. Explain 3 steps a community can take to reduce water pollution. 3 steps that a community can take to reduce water pollution. Yeah, like the water carriage. You all got that right. But all of you are urban citizens. You don't want to read about it, pit toilet. So none of you know about pit toilet. When you get to the exam, you can't answer how it works, where it should be constructed. And I have a question on the 2020 paper. Beast that you can't answer that question. You can't answer the one on the 2020 either. All right. So you urban folks, you need to go and read about pit toilet that your grandparents used to use, and some of us still use it. All right. It is something that we need to address. So that was the 2017 that we were excited about. I am going to do the 2020 now, then I'm going to do the 2018. All right, so you know where your weakness lies. Your weakness is definitely in the pit latrine. You need to look at that. So we are going to 2020, and I like the 2020 paper. One thing about the 2020 paper is that don't worry yourself, just read and make sure so you have it locked for example. It's going to be question number 5 or number 6. We're section A of the syllabus now. We are on the 2020 past paper. We are doing May, June 2020, and we are going to go through this very, very quickly. Please listen to the question very carefully. List 4 characteristics of living things. 4 characteristics of living things. Type them one time. Let's go. 4 characteristics of living things. Type it in the chat. 4 characteristics of living things. Four characteristics of living things. Remember, your exam is 1, so if you need to travel to your exam, or if you're going to get a taxi, you need to make arrangement to get to that cab, so that cab to get to you. You really don't want to be late for the exam because you're here with Mr. Wilson. Movement, excretion, nutrition, and growth. Excretion, respiration, locomotion, reproduction. Excretion, respiration, nutrition, movement. Movement, irritability, growth, reproduction. Growth, respiration, excrete, nutrition. All right, there we have it. Great. Those responses were awesome. State 2 organelle found in a plant cell which are not found in the animal cell. 2 organelle in a plant cell that is not found in the animal cell. 2 organelle in a plant cell that is not found in the animal cell. I don't know why we have stimulus here, any at all. I don't know why we have store water, any at all. Vacuole is wrong. Cytoplasm is wrong. Guys, I want you to listen. Whether the vacuole is large or small, listen to what the question asks, because if you write that, it is wrong. The question is asking you, state 2 organelles. A vacuole is a vacuole, whether it is large or small. So state 2 organelles, you have to listen to how the question is coined. State 2 organelles found in a plant cell which are not found in animal cells. You can't use vacuole. While it is true in some cases, it's not true now. So all of you should be just jumping out. Cell wall, chloroplast. That is everybody should be saying the same thing because it's the easiest thing to remember. Cell wall, chloroplast. And now that I see that some persons are struggling, tell me one organelle that is found in the animal cell that is not found in the plant cell. One organelle in the animal cell that is not in the plant cell. Yes. One organelle, thank you very much. Glycogen granules are found in animal cells, but they are not found in plant cell. Starch granule instead are found in plant cell. Very good. I have to rough you up. Ribosome is found in both of the cells. Here, ribosome is for protein synthesis, so don't get that wrong. Let us move on. Yeah? No, I'm not upset. I'm not upset. I'm sort of excited, not upset. All right. Sanjay carried out an experiment to confirm that oxygen is generated from photosynthesis. All right. So we're looking at oxygen being, I'm too excited. I think I need to calm down. All right. He carried out an experiment. Let me show you the experiment. All right, great. That is the experiment. And the video, there's an animation on my channel for this. All right, so Sanjay carried out an investigation to confirm that oxygen is generated from photosynthesis. All right, we're seeing that. So what are we supposed to do? He followed the procedure below. So this is the procedure below. I'll read the procedure for you. It says here, count the number of bubbles generated in experiment A and count the number of bubbles generated in experiment B. Remove the test tube in experiment A by placing your finger over the mouth, taking it out of the water and turning it over. Move your finger and place a glowing splint into the tube and observe what occurs. Repeat the step for experiment. Repeat the step 2 to 3 times for experiment B. All right, one thing we're looking at photosynthesis here. And remember, photosynthesis is fueled by the light energy. So here we have the light energy from a lamp. It is pretty much similar to the light energy from the sun. Let us move on to see what happens on this side. You are supposed to state which experiment A or B would generate more bubbles. So looking at A or B, A has a lamp beside it. Let me just see if there's any other thing for us to look at. All right, great. So A has a lamp beside it, while there's no lamp beside B. Now, which of the experiment, oh wow, which of the experiment A or B would generate more bubbles? Yes, it is going to be A, and it's going to be A because more oxygen is being released because the light there powering photosynthesis. Very good. The other question is going to be state what Sanjeev would have observed happening to the glowing splint in the experiment in A and in B. What would happen to the glowing splint? What would happen to the glowing splint? All right, Ventura, I note your language. The glowing splint would glow brighter. All right, great. So remember, a test for oxygen is that oxygen relights a glowing splint. So if we have a lot of oxygen gas in A, the chances of that gas relighting that splint is greater. Now, there's of course going to be some amount of oxygen in B, but it might not relight the glowing splint, but the splint will continue to glow. But in A, we're almost certain that the splint will be, of course, reignited. All right, so again we go. This one here says, identify the gas that could have produced the bubbles in experiment B and state, something is wrong here. Identify the gas that would have produced the bubbles in experiment B and state the process which would have produced this gas. I think I want to look back at that question. I don't know why they would go here with this. Is it not the same process that identify the gas that would have produced the bubbles in experiment B and state the process. Okay. What's the gas? What's the gas for experiment B? It's going to be oxygen. No, the process can't be combustion. If it is combustion, it is of course going to be carbon dioxide. So I think that both tubes will be producing oxygen, and the oxygen is as a result of photosynthesis. But if we are going to be looking at the splint burning, then it's going to be combustion causing the splint to burn, and then carbon dioxide will be given off from the splint. All right? There are two different things we're looking at here. Now, explain why Sanjeevs can conclude that oxygen is a byproduct of photosynthesis. Why is it that he can conclude that oxygen is a byproduct of photosynthesis? Why is it that he could conclude that oxygen is a byproduct of photosynthesis? Type it in the chat for me very quickly. Where are we now? Because the water. Oh Lord Jesus, you're gone far, Ventura. All right, so let me just help you with that. The question says, explain how Sanjeev can conclude that oxygen is a product of photosynthesis. Let me explain that very quickly for you. Now, the setup that we have here is that you load a plant usually in a glass funnel and the light. Now, while photosynthesis takes place, the gas is going to be collected at the end of the tube. Now, if the gas that is in the tube would have caused a glowing splint to relight, then we can say that oxygen is given off as a byproduct because oxygen relights a glowing splint. So the gas that was in the tube is actually oxygen because it actually relit the glowing splint. Right? So that is the conclusion. Conclusion, the gas in the tube, which is a product of photosynthesis, relit the glowing splint, and that is pretty much a positive test for oxygen. All right, great. What's the other question now? State which experiment served as a control and give one reason for your answer. Which of the experiments served as a control? Which of the experiments served as a control? Control, you don't know what's a control. A control is a variable in an experiment that you know how it operates, you know what the end result is supposed to be, right? So it is somewhat unaltered because you are using it to judge whether or not the outcome of the experiment is going to be true. So in this case, you'd have to look at the aim. It says that carried out to investigate or confirm that oxygen is generated from photosynthesis. So our control here is going to be B. For me, it's going to be B because we knew that the plant would have produced some amount of oxygen. We know that if we increase the light, the oxygen would have increased. So experiment B, the addition of light from the lamp would change the outcome. I don't know what this is saying. For me, the answer is going to be B. That's the control because we added the light to the other one and we did see that oxygen is produced. Which experiment? Control. I have B as a control. I have B as a control and A as the real experiment. That's for me. That's my take on it. All right, so let's just move on. All right, this question is on meiosis. And we're at question number 4. Question number 5 is excretion. And question number all right, please watch your time. I'm trying to just get there. Question number 6 is on pollution. So 2020 paper is a nice paper. Most of what you are going to be getting today should be from this paper. All right. So I wanted to release you at, all right, come. Cell division promotes growth and development in organisms. These processes occur by mitosis. Identify one cell in the body in which mitosis takes place, in which mitosis takes place, and one cell in which meiosis takes place. Let me say that in the chat now. If you let me down this morning, Simon, one cell in the body in which mitosis takes place, and one cell in the body in which meiosis takes place. All right, this is like an easy genetic question. Guys, don't complicate this question, man. Mitosis take place in muscle cell. Any cell at all you name in the body, other than sperms and egg, mitosis take place in. Meiosis only take place in the sperm and egg cell. Meiosis only takes place in the sperm and egg cell. All other cells is mitosis. Let's move on. All right, so figure 5 shows the process of meiosis, and you are supposed to, in each labels, match each label, state of the cells above with the description below by placing the correct letter A, B, or C on the line next to it. All right, so you remember we spoke about the 3 words we are going to be using: DNA duplication. We are going to be using haploid, and then we are going to be using, great. So I'm going to show you. Here we have A, B, and C. So this is A, B, and C. Hope you see it. A, B, C. A, B, C. Great. This is your question now. What is A? Is A haploid condition or a diploid condition? What is A? Is it haploid or diploid? So you have it that A is diploid. I'm listening to everyone. What is A? Is it haploid or diploid? Haploid, diploid. What is B? Is it haploid, diploid, DNA duplication? What is your answer? All right, sorry guys, sorry. All right, so you are saying that, all right, for me, I can't see the diagram. So I have answers on the paper, but I can't say that it is really, it's not copied properly for me. But my answer on the paper says that C is actually haploid. A is actually diploid. And C must be haploid because C is the end result. So it must be haploid after the number there. And of course, naturally, B is going to be the duplication, and A there being the diploid number, the full number. All right, that's what I have for that. All right, so the next question is going to be state one factor that would account for the variation in daughter cells at the end of meiosis. One factor that could account for the variation. One factor that could account for the variation. Remember that I gave you a number of factors that could cause variation. But at the end of meiosis, what does that mean that could cause variation? No. I showed you when I use my fingers and put them together. What is that process called? Let's use that one. Crossing over. Crossing over. Prophase 1, crossing over. Crossing over in prophase 1. All right, not mutation. Crossing over in prophase 1. Now, the other question, this paper, this 2020, looks so much like what you're going to do today. All right. In prophase 1, we were talking about Hitchhiker's thumb earlier. So this is it. In this question, this is it. So Hitchhiker's thumb versus your regular stuff. All right, figure 6 and figure 7 illustrate a straight thumb and a Hitchhiker's thumb. All right, listen. The allele for Hitchhiker's thumb is recessive. So Hitchhiker's thumb is a recessive condition. And the allele for straight thumb is dominant. So use a capital letter. Barry and his father both have Hitchhiker's thumb. They both have Hitchhiker's thumb. Now remember what we said about a recessive allele or a recessive condition. If it's a recessive condition, then you must acquire both common letters in order for the condition to be shown. You must have two small letters. So it must have to be two small h if you have the disease in this case, and if you don't have the disease, it's going to be not disease. If you have the Hitchhiker's thumb, you are going to have two common H, but if you have a regular thumb, you are going to have two capital H. So this is the question. Identify the genotype for Barry and his father. What is the genotype for Barry and his father? What's the genotype for Barry and his father? The genotype for Barry and his father? No, remember the allele that we are using is H, you know. So what's the genotype there? We are using H, the letter H. Happy? H. Remember, the rule says about inheritance of recessiveness. Once you have the condition, you have two small letters. I'm making it very simple for you. So if both persons have Hitchhiker's thumb, then both have the homozygous recessive allele, which is two small h. All right, great. What is the mother's genotype? Now, this is going to be interesting. The mother genotype, I think, could be two things. She could be heterozygous as well as she could be homozygous recessive. We don't know, but in order to get the offspring to have the recessive allele, she would have had to have the recessive allele at some level. It's either she have it as a carrier, where heterozygous, or she have it in the homozygous recessive state. So I say everybody putting up HH, which means that you guys have a copied paper. But it could be anyone. The mother couldn't be, it could be either one. Right? It's either it's capital H, common H, or the mother has two common H. Because it didn't say what the mother looked like. It just tell us about the father. It just tell us about the father and the offspring. Right? So if the father have the whole dominant stuff, then the mother could not be dominant. Because if the mother was dominant, then it is impossible for the offspring to have the ichicotome. If the mother was dominant, it would have been impossible. All right? Great. So always remember that. There are two possibilities for mother. For the father is going to be like the son, is going to have too small h. And they were asking us to put it in a Punnett square, but you guys solved it long before we got there. But they asked us something else. They say Barry mother is now pregnant. Complete a Punnett square below and determine the likelihood of her unknown child, the likelihood that her unknown child will have ichicotome. Now we have to take a position. It's either you are going to have her as heterozygous or you are going to have her as homozygous recessive. And then you are going to do that cross with the father, because you know what the father is. Depending on whether you have the mother as heterozygous or you have the father as homozygous, that will determine the chances of your result. All right. So I won't touch that because we could go either way. So if we use the mother as heterozygous, the answer is going to be that there's a 50% chance that the offspring will have ichicotome. If we use the mother as being heterozygous, there's a 50% chance that the offspring will have ichicotome. And then the other question here said figure 8 illustrate data obtained on a figure 8 illustrate data obtained on the angle at which persons with ichicotome can be bent. So persons with ichicotome, they have this data. Without answering the question, you shouldn't do this in the exam. Once you hear that it has the angle to which ichicotome can be bent, what comes to your mind? What comes to your mind? Type in the chat. We are coming down. We are coming down, down, down. We are coming down. Oh, with the glory of the Lord, we are coming down. No. Hey, what will not tell me, say? What must come to your mind is that it is a continuous variation, because we are talking about varying degree in which it can bend. Come on, man. Apply that thinking skill. So let us look and let me see where it is going. Then look, we have this nice curve histogram that pretty much shows the thing. So let me see where they are going with this. Identify the type of, there we go. Identify the type of variation characterized by the data given. Now look at the data that is given. One, two, three, four, five, six. And we are looking at the varying degree. So some person can go 1 degree, 2 degree, 3 degree, 4 degree. All right. So it must be continuous variation. Another thing, based on the type of variation identified in C, state whether genetic or environment or both determine phenotype of hitchhiker? What determines the hitchhiker? How far back the finger can bend? All right, so we have done excretion already. So the question might be a little different. So I'm just going to jump on into pollution and jump the excretion question here. We said genetics are, it's going to be a little bit of both, because if I continue to press my finger backward, after a while, the degree of the hitchhiker thumb might change. Like some persons can't do that. But after a while, when you hold your finger and you're practicing doing that, after a while, you can do it. All right, so it's going to be both. All right, so I'm going to jump over for the sake of persons are exhausted now. We have reached a threshold, which is 11:30. You need an hour and a half there about to go and check your friend, to travel, to move to exam. So I am not going to be getting into the 2018 paper today. I am sorry. It doesn't have much question on it though. I'm going to be doing the pollution paper. I'm going to be sending you off, wishing you all the best. And probably I see you this evening with the paper. Probably I see you this evening with the paper. All right. So I am packing up. This is the end of 2020, 2021, the COVID online teaching. Remember to like, share, and subscribe, and click that notification bell when you click subscribe and select all, so that you'll be able to follow what we are doing. You are going to be able to share what we are doing. You are going to be able to share the success story, or you got a 1 for HSB. You are going to be leaving that on the channel. I'm going to go on the road after this question. I'm going to go to the exam later this evening. It's a 1 o'clock exam, so I might get there about 2:30 thereabout. And I'll try to get a copy of the paper. The paper is free after the exam, the paper 2 only. So I'll get a copy of the paper and I'll see how well you did. We are going to try to see if we can answer that paper and see, you'll see how close you came to it. But remember, you're all going to do well. But if you didn't do well, or if you don't do well, you can always come back to us. But we'd prefer if you don't come back to us to do HSB again. We'd prefer you come back to tell us about the grand story. All right. So the first thing here is to define the term pollution. What's pollution? We did that this morning. What's pollution? What's pollution? Pollution is the introduction of contaminant to the environment. Pollution is the introduction of contaminant to the environment. Pollutant is the contaminant that would have been introduced to the environment. State 2 examples of air pollution other than carbon dioxide. You have particulates, you have nitrous oxides, you have carbon monoxide, you have, did I say methane? You have chlorofluorocarbon. All of these are examples of pollutant to the air. Yes, smog, all of that. Oh, you guys know. I thought you didn't know. All right, great. So you're doing well. All right, all of those. Guys, remember traffic. Traffic can be on the road, so you want to cut out early. Identify 2 resources, 2 sources of air pollution. 2 sources of air pollution: exhaust from motor vehicle and smokestack and volcano. Those also contribute to air pollution. All right. The car sources, where it comes from. So it comes from the exhaust from burning fossil fuel, from burning any fire, from the smokestack. All those contribute. Factories, right? Factories and exhaust pipe. Great. Here we are asked to do an analysis. So it says that figure 9 depicts the percentage of carbon dioxide produced by yearly transportation activity of residents in a town. Now you are expected here, it says that persons on vacation traveling, 49%, those who are on vacation traveling, 49%. Hometown travelers like me, 19%, and daily commute, 32%. 32%. This is what we're supposed to do. Most residents go on vacation once a year, but commute to work or school approximately 5 days a week. Explain why vacation traveling produce significantly more carbon dioxide than daily commute, and suggest 2 ways in which this amount of carbon dioxide produced from the daily commute can be reduced. What do you think? Why is it that persons on vacation is pretty much contributing more carbon dioxide than persons who commute to work, despite persons who commute to work go to work 5 days per week? Let me have that. And then after that, we have one more question, and that's the end of the 2020 paper. Because everyone will be taking individual transportation. That is the first thing. Everybody boasting and I go show off, yo, me think shot, you know, some they are firing and me a chopper and all of them stuff there. And everybody will be driving their own cars to work or their own cars around. They'll make 50 trips on the road that all the girls can see them and all the boys can see them. All right? They are more round and about. They want everybody to see them. Hey, I am over here. You can't see me. Hey, look, I'm going to rev up the engine. Right? And most time when persons are on vacation, they can't drive. They can just barely steer the vehicle. So they're going to press that exhaust more than they should, emitting more carbon dioxide into a more pollutant, carbon dioxide as it is now, into the atmosphere. How can we correct this problem? We can reduce it by carpooling and riding a bicycle. Great. We can reduce it by riding a bicycle and carpooling. Last question, and then you go. All right? Many Caribbean countries have banned single-use plastic items, such as straw and plastic bags, outline 2 reasons why this is a good decision for marine ecosystem and the wider community. 2 reasons. That takes us to the end of May, June 2020 paper. 2 reasons why it's a good reason. Two reasons. We are at the end of the session. It was nice having you. I have done what I've not done for ages. I had this awesome opportunity to work with students from right across the Caribbean. And it was indeed a treat. It was a treat for my team and I. We saw persons grow from sending us a message every second to start answering questions in class. That is indeed true growth. And we have persons who, when they started out, they said that we're not going to be doing the exam because we don't know what we are going to do on the paper. To persons, some of those persons now saying that I am going to be getting a 1. Some of these persons are so confident that they are already on their way to the exam, and there's nothing else for them to add to their repertoire or to that reservoir, of knowledge. Now, we know that the plastic and all of those stuff, they stifle organism in the water. We did this last night. They cause organism to be deformed. They kill they kill life forms and all of that. We answered this question last night. So I am going to wish for us all the best. I know that things are going to work out. I know that CXC is pretty much concerned about you, and they are going to give you a paper today that you are going to say, whoa, I over-prepared for the exam. But whether you over-prepare or under-prepared, make sure that you come out of the exam on top. To the following countries who were a part of our session, we say thanks. Jamaica, Trinidad and Tobago, Guyana, Barbados, Grenada, St. Vincent, Antigua and Barbuda, St. Kitts and Nevis, Dominica, St. Vincent and the Grenadines, United States, Cayman Islands, Anguilla, Belize, St. Martin, Zambia, British Virgin Islands, India, Turks and Caicos, Venezuela, Canada, Brazil, Suriname, to all these countries from which students would have logged in, we say thanks to you for joining, and we wish for you all the best. For all the parishes in Jamaica that were represented, we say thank you and all the best. For all the schools that were represented, we say thank you and all the best. We encourage you to share the message with those students in lower school so that they can join us early enough so that we can increase or improve the performances in science across the Caribbean. We continue to prepare animation notes lab for students in the Caribbean and to pretty much present the content at the level and diction of persons of the Caribbean. We continue to look at the CXC syllabus when we prepare our content and, of course, the syllabus or curriculum of lower school. We have been looking at sciences at lower school and we have been working at animation to make science be more fun for students at lower school. We do ask where it is possible for persons to partner with us that we can continue to reach the fortunate and the less fortunate. Our mind or our memory bring us back to persons who we serve who reached out to us during the 1 year of COVID lockdown. We recall a student in the United States who visited for a surgery and was there for quite an awesome period, could not make it back home because of COVID. We recall a young lady from St. Elizabeth saying, "Sir, the only time I have access to class is on the weekend when I travel to my aunt on the lower grounds." We recall a young lady that was trapped in Trinidad and Tobago and could not go back home because of COVID. They were all with us when they could. We recall the many persons who would have told us that they have been in disconnect with schools because schools would have been closed and the technology was a remote possibility for many persons and many schools. I am grateful for all those who extend the assistance. We were able to garner past paper from teachers from right across the Caribbean. We were able to have the support from students from right across the Caribbean. There are other vloggers who reached out to us and encouraged us to work. Vloggers would include persons like Michelle, CSEC biology tutor, Karen Springer, Matt's there from Trinidad. We had conversation. Miss CSEC biology and chemistry. These are just some of the many persons who reached out to offer support. I want to say thanks to Miss Shernich White from Torrent High School. I want to say thanks to my teacher there from Kellitz High School, another teacher who would have helped us, forgive me, I don't have the listing of the names here, from Porus High School, and of course to Miss Chanel A there from Brownstown, Mr. Arville McLeod there in the USA. From time to time, our research in presenting would have taken us more than just the syllabus. We would have had teachers who are lending their support. I want to say thanks to my own team at work. I want to say thanks to my own team at work who would have allowed me some amount of space to prepare students for biology, which went on Monday, and the students today for HSB. I am most grateful. I must also say thanks to my family who made the time available for me to be with you. It was a sacrifice. From time to time, and I came by just to say I, or to shove a desk or a chair to say I am here and make sure you're doing well, Daddy, because I'm looking on. To my extended family who logged on from various places in the world to say that do this, do that, change this, change that, we are indeed grateful. We plan to continue to serve, but of course, we would love to have an assistant to make this a reality come next year. We look forward for those wonderful comments, the passes that will come from CXC, and we will continue to be at our best in serving the nation, in serving the Caribbean, in serving the world, and make it be a beautiful place to continue to live. We wish for you all the best in your exams. Take care.